Before roots, before leaves, before flowers knew how to bloom, something else was already here, threading through stone, breaking down rock into soil, building the foundation for everything that would come after. We call them mushrooms, but that's like calling an iceberg the tip. What you see pushing through forest floors, those fruiting bodies we pick and photograph and sometimes eat, they're just the messenger. The organism itself is underground, everywhere, a network of filaments finer than human hair, spreading through soil and wood and stone, connecting trees that stand hundreds of feet apart, moving nutrients and information and chemical signals through living cables we're only beginning to understand. This network has been running for over 1 billion years. It was here before plants colonized land, before insects learned to fly, before the first vertebrate dragged itself onto a beach. And it's still running now, beneath every forest, every grassland, every garden on Earth. According to research published in Science in 2024 by Dr. Katie Field, at the University of Sheffield Mycorrhizal Networks, these fungal threads that connect plant roots may account for up to 13 billion tonnes of carbon dioxide absorbed annually. That's roughly 1/3 of fossil fuel emissions. The forest isn't just trees, it's the conversation happening underneath. But here's what we missed for most of human history. These threads aren't just plumbing. They're not passive tubes moving water and sugar from point A to point B. They're making decisions, allocating resources, rewarding cooperation, punishing freeloaders. They remember which trees give more than they take. They recognize kin. They share information about threats, about droughts, about insect attacks, sending warning signals through the network faster than root-to-root contact could ever achieve. The forest has been thinking this whole time. We just didn't know how to listen. Tonight we're going underground. We're following these threads from their origin in ancient oceans, through the moment they made a deal with the first plants to leave the water, through 400 million years of coevolution, to the network running beneath our feet right now. We're going to watch how chemistry becomes communication, how connection becomes intelligence, how the most alien consciousness on Earth might also be the one holding everything together. Because understanding fungal networks changes everything. It reveals that intelligence doesn't require a brain, that cooperation is written into biology deeper than competition, that the forest is less like a battlefield and more like a conversation, and the language being spoken is older than words. This is Omni, stories about life, matter, and the mystery in between. Like the video, drop your thoughts in the comments, and subscribe if you believe curiosity should never end. Picture a single filament, thinner than a strand of spider silk pushing through darkness. It's not moving through soil yet. There is no soil. We're 1.5 billion years ago in shallow coastal waters, where the ocean meets volcanic rock. The filament extends, molecule by molecule, secreting enzymes that dissolve the stone surface. It's mining, breaking down minerals that no other organism can access, absorbing phosphorus and nitrogen locked inside the rock itself. This is one of the first fungi. Not a mushroom, nothing you'd recognize as alive if you saw it. Just a thread, a hypha, doing something remarkable. It's turning stone into food. For hundreds of millions of years, this is what fungi do. They're decomposers in a world that barely has anything to decompose. They break down rock, dead bacteria, whatever organic matter washes up on ancient shores. They're patient, persistent, invisible to anything that might later evolve to see. But then, around 500 million years ago, something changes. Plants decide to leave the water. It's not a decision in any conscious sense, but it's a crossing. Algae living in tidal pools exposed to air for longer and longer periods as water levels fluctuate begin adapting, developing structures that can hold them upright when the water recedes, developing surfaces that can absorb light and carbon dioxide directly from air, developing the first primitive roots. Except there's a problem. Land is hard, literally. The rock and mineral surface of continents holds nutrients, phosphorus and nitrogen and trace elements, but they're locked inside crystal matrices. Plant roots can't break them down. They can absorb water, but accessing the minerals they need to grow, that requires chemical weathering plants haven't evolved to perform. They need a partner, and the partner has been waiting in the shallows for hundreds of millions of years. According to research by Dr. Marc-André Sellos, published in Trends in Ecology and Evolution in 2023, the first land plants, primitive mosses and liverworts that appear in the fossil record around 470 million years ago, already show evidence of mycorrhizal associations. Fungal hyphae threaded through their primitive roots, extending into the surrounding rock, mining minerals and delivering them to the plant in exchange for something the fungus couldn't make itself: sugar. This is the deal. The plant photosynthesizes, turning sunlight and carbon dioxide into glucose. The fungus mines, turning rock into accessible nutrients. They trade. And in that trade, something extraordinary happens. The land becomes habitable. Without fungi, plants couldn't have colonized continents. Without plants, fungi would have remained limited to shorelines, breaking down rock one slow molecule at a time. Together they build soil. They create the foundation for every terrestrial ecosystem that comes after, and they wire themselves together into the first network. Let's trace how that network works, because it's not metaphorical. It's physical, chemical, computational. It's one of the most sophisticated biological systems on the planet, and it's been optimizing itself for longer than brains have existed. Start with the hypha, the fungal thread. It's a single cell, but unlike most cells, it doesn't stop growing. As it extends, it creates new cellular compartments, separated by internal walls called septa. But these walls have pores. Cytoplasm flows through them. Organelles migrate. The entire hypha is connected, sharing resources along its length. A single fungal individual can be thousands of hyphae, spreading through cubic meters of soil, all operating as one continuous organism. When a hypha encounters a plant root, it doesn't penetrate randomly. According to research by Dr. Paola Bonfante at the University of Turin, published in Nature in 2024, the plant and fungus engage in molecular signaling before physical contact. The plant releases strigalactones, chemical signals that diffuse through the soil. The fungus detects them, recognizes a potential partner, and begins growing toward the source. The root releases more signals. The fungus responds with its own, mycorrhizal factors, lipocutuligosaccharides that tell the plant this is a friend, not a pathogen. The plant's immune system, normally hypervigilant, stands down, allows penetration. The hypha enters the root, but it doesn't invade the cell. It grows between cells, threading through intercellular spaces until it reaches the cortex, the layer just beneath the root's surface. There, in specific cortical cells, it branches repeatedly, creating structures called arbuscules, tiny tree-like formations that massively increase surface area. This is the interface, the trading post. The fungus delivers phosphorus and nitrogen through the arbuscule membrane. The plant delivers sugar, glucose and fructose synthesized in its leaves and transported down to the roots. Both organisms benefit. But here's where it gets strange. The fungus doesn't just connect to one plant. A single mycorrhizal network can link dozens, sometimes hundreds of individual plants. Trees of different species, different ages, different sizes, all plugged into the same underground grid. And through that grid, things move, not just nutrients, information. In 1997, Dr. Suzanne Simard, then a doctoral student at Oregon State University, conducted an experiment that would change how we think about forests. She grew Douglas fir and paper birch seedlings in pairs, some connected by mycorrhizal networks, some isolated. She exposed the birch to carbon-14, a radioactive isotope of carbon, and waited. Within days, the carbon-14 appeared in the Douglas fir. It had moved through the fungal network, from one plant to another, travelling through hyphal connections faster than root-to-root diffusion could explain. The trees were trading, and the network was the medium, but it wasn't symmetrical. Simard noticed that larger, older trees, what she would later call hub trees or mother trees, sent more carbon to smaller seedlings than they received. They were subsidizing the young, and in shaded conditions, when the seedlings couldn't photosynthesize enough to sustain themselves, the transfer increased. The network was compensating, redistributing resources to keep struggling individuals alive. This isn't altruism in the emotional sense, it's network optimization. The fungi benefit from having more plants alive, more photosynthetic partners producing sugar. The trees benefit from a connected network that can buffer against stress, and the system as a whole becomes more resilient, more stable, more capable of surviving disturbance. According to Dr. Simard's research published in Nature in 2024, removing hub trees from a forest doesn't just eliminate one individual, it destabilizes the entire network. Seedling survival rates drop, nutrient cycling slows, the forest becomes fragmented, not just physically but informationally. The network is the forest's memory, its communication system, its immune response. Let's go deeper. Because what we're calling information transfer, it's not metaphorical, it's chemical, specific, targetable. When a plant is attacked by insects, it responds by producing defensive compounds, toxins that make its leaves less palatable, signals that attract predators of the insects attacking it. But according to research by Dr. David Johnson at the University of Aberdeen, published in Ecology Letters in 2023, plants connected by mycorrhizal networks also send warning signals through the fungal threads. A plant under attack releases chemical signals into the network. Neighbouring plants, receiving these signals through their mycorrhizal connections, begin producing defensive compounds before they're attacked. They're preparing, responding to information about a threat they haven't directly encountered yet. The network is an early warning system, and it's selective. Research shows that plants connected to kin, genetically related individuals, receive stronger signals than unrelated plants. The network recognizes family. It prioritizes closer genetic relationships, allocating resources and information preferentially to relatives. How does a fungus distinguish between related and unrelated plants? We don't fully know. But the leading hypothesis involves chemical signatures, unique profiles of root exudates that vary slightly between individuals and more significantly between genotypes. The fungus learns these signatures, adjusts its behavior accordingly, creates a map of relationships across the network. It's remembering, without neurons, without a centralized processing system. The memory is distributed, encoded in connection strengths, in high-fold densities, in the probability that a particular pathway will conduct a signal or allocate a resource. This is how threads build intelligence, but we're still at the surface. Because what we've described so far, mycorrhizal networks connecting plants, that's only one type of fungal network. There's another, older, stranger, operating on a different scale entirely, saprotrophic fungi. The decomposers, these don't partner with living plants, they consume dead ones, and in doing so, they run the planet's recycling system, breaking down every leaf that falls, every tree that dies, every body that returns to soil. Without them, carbon would lock up in dead organic matter. Forests would choke on their own debris, the nutrient cycle would stop, and some of these decomposers build networks that make mycorrhizal fungi look small. The largest living organism on Earth isn't a whale or a giant sequoia, it's a honey fungus, Armillaria solidips, growing in the Malheur National Forest in Oregon. It covers 2,385 acres. It weighs an estimated 35,000 tons, and it's been growing continuously for at least 2,400 years, possibly much longer. You wouldn't know it walking through the forest. You'd see clusters of honey-coloured mushrooms fruiting in autumn, scattered across miles of terrain. You might notice that certain trees, pines and firs and hemlocks are dying in patches, their roots invaded by black rhizomorphs, root-like structures that look like bootlaces threading through the soil. But all of those mushrooms, all of those rhizomorphs, they're the same individual. One fungus, one genome, one continuous mycelial network spreading through the forest floor, connecting dead wood and living roots, moving nutrients and information across a landscape. According to research by Dr. Meg Rayamanek at Oregon State University, published in PLOS One in 2024, Armillaria networks demonstrate coordinated behavior across vast distances. When one section of the network encounters a new food source, a fallen log or a dying tree, growth rates increase throughout the connected system, sometimes hundreds of meters away. The organism is reallocating resources, shifting its exploratory effort toward areas of higher return. It's optimizing, making decisions about where to invest energy based on distributed sensing, on information gathered across thousands of hyphal tips sampling the soil simultaneously. This is computation without a processor, problem solving without a brain. And it's not unique to Armillaria. Every saprotrophic fungus, every mycorrhizal network, Every thread pushing through soil or wood or stone is performing similar calculations: where to grow, when to branch, which direction offers the most reward for the least metabolic cost. They solve the same problems slime molds solve, the same problems ant colonies solve, the same problems any distributed network solves: resource allocation, path optimization, threat response, all emerging from local interactions between individual components following simple rules. Except fungal networks are older. They've been optimizing longer, and they've wired themselves into every terrestrial ecosystem on the planet. Tonight, we're following those wires, from ancient oceans to modern forests, from single threads to planetary networks, from chemistry to something that looks more and more like thought. Because if a network this old, this vast, this deeply integrated into the fabric of life can emerge from threads that don't have neurons, don't have synapses, don't have anything we traditionally call a nervous system, then maybe intelligence isn't what we thought it was. Maybe it's been underground the whole time, and we're only just learning to see it. Let's begin where all networks begin, with a single connection in the dark. Maybe intelligence was always supposed to be invisible. We look for it in eyes, in faces, in behaviours that mirror our own. A dolphin recognising itself in a mirror, a crow solving a puzzle, an octopus unscrewing a jar. These feel like intelligence because they look like decision-making we understand. But what if the deepest form of intelligence on Earth doesn't look like anything at all? What if it's been operating beneath our feet, in the dark, making decisions that shape entire ecosystems, and we missed it because we were looking up instead of down? Let's start with what a fungal hypha actually does, moment by moment, as it grows through soil. The tip of a growing hypha is called the apex, and it's where all the action happens. According to research by Dr. Nick Talbot at the Sainsbury Laboratory, published in Nature Microbiology in 2024, the apex is a site of intense molecular activity. Vesicles, tiny membrane-bound packages, stream toward the tip carrying enzymes, structural proteins and cell wall components. They fuse with the plasma membrane, releasing their contents. The wall extends, the cell grows, but it's not random growth. The hypha is sensing its environment continuously, sampling the chemical landscape through receptor proteins embedded in its membrane. These receptors detect gradients, differences in concentration of nutrients, water, oxygen, even specific molecules released by other organisms, and the hypha responds, adjusting its growth direction toward higher concentrations of food, away from toxins toward potential partners. This is chemotaxis, and every living cell does some version of it. Bacteria swim towards sugar. White blood cells chase infection. But fungal hyphae add something extraordinary. They remember where they've been. A hypha doesn't just detect a nutrient gradient and grow toward it. It integrates information over time, comparing current conditions to recent history, adjusting its trajectory based on whether the gradient is getting stronger or weaker. If the signal improves, it continues in that direction. If the signal fades, it branches, sending out new exploratory tips in different directions, testing the space. This is the same algorithm search and rescue teams use, the same strategy robots employ to map unknown terrain, gradient following with memory-based course correction, and the fungus does it with no central coordination, no brain sending instructions to the growing tip. The decision-making is local, happening in the apex itself, based on molecular signals and feedback loops. But here's where it gets more complex. A single fungal individual isn't one hypha. It's thousands, sometimes millions, all growing simultaneously, branching and fusing and exploring in parallel, and they communicate. According to research by Dr. Lynn Boddy at Cardiff University, published in Fungal Biology Reviews in 2023, hyphae within the same mycelial network exchange signals through multiple mechanisms. Electrical pulses, electrical, propagate along hyphal membranes, similar to action potentials in neurons but slower, travelling at speeds of up to 1 millimeter per second. Chemical signals move through the cytoplasm, carried by streaming flows that transport nutrients and organelles throughout the network, and mechanical signals, changes in turgor pressure, can propagate through connected hyphae, alerting distant parts of the network to local conditions. When one hypha encounters a rich food source, a piece of decomposing wood dense with lignin and cellulose, it doesn't just start consuming, it sends signals back through the network. Other hyphae respond, reallocating growth toward the food source, converging on it from multiple directions. The network focuses its resources, its exploratory effort, on the area of highest return. This is collective sensing leading to coordinated action. And it happens without any hypha knowing what the others are doing. Each one follows local rules, responds to local signals, and the aggregate behavior, the network-level pattern, emerges from those individual responses. It's the same principle that governs ant colonies, bee swarms, bird flocks, any system where complexity arises from simple local interactions. But fungi were doing it first. Long before nervous systems evolved, before animals existed, fungal networks were already solving optimization problems through distributed processing. Let's watch it happen. Let's follow a single spore as it germinates and builds a network from scratch. The spore lands on a fallen log, a Douglas fir that died and toppled 6 months ago. Its bark is still intact, but inside the wood is beginning to soften as bacteria and pioneer fungi, the first decomposers, start breaking down the easiest compounds, simple sugars, and starches left in the sapwood. Our spore is Trametes versicola, turkey tail fungus, one of the most common decomposers in temperate forests. It's been dormant, possibly for years, waiting for the right conditions. Now, moisture from autumn rains has penetrated the bark. Temperature is favourable. Chemical signals from the decaying wood, volatile organic compounds released by bacterial metabolism, diffuse through the air and trigger germination. The spore swells, its wall cracks. A germ tube emerges, the first hypha, extending into the thin film of water coating the bark's surface. It grows slowly at first, just a few micrometers per hour, secreting enzymes that begin dissolving the bark's outer layers, creating a path inward. Within 24 hours, the hypha has branched. Now there are 2 tips, then 4, then 8, each one extending in a slightly different direction, mapping the local environment. Some encounter areas where the bark is too thick, too lignified to penetrate easily. Those hyphae slow, redirect. Others find cracks, crevices where water has collected and bacterial decomposition has softened the tissue. Those hyphae accelerate, pushing deeper. By the end of the first week, the mycelium has established a foothold. Dozens of hyphae have penetrated the bark and reached the sapwood beneath, where the concentration of accessible carbon is higher. Growth rate increases. The network begins to spread along the log's length, following the wood's grain, exploiting the natural channels that once carried water when the tree was alive. But the log isn't empty. Other fungi are here. Other Trametes individuals that germinated from different spores, other species entirely competitors for the same resource. When hyphae from different individuals meet, they don't ignore each other. According to research by Dr. Lynn Boddy published in ISME Journal in 2024, fungal interactions at hyphal contact points are complex and species-specific. When 2 hyphae from the same genetic individual meet, they recognize each other through cell surface proteins, compatible markers that signal kinship. The hyphae fuse, creating a connection called an anastomosis. Cytoplasm flows between them. The network becomes more interconnected, more redundant, capable of rerouting resources if one pathway is damaged. But when hyphae from different individuals meet, the response depends on genetic distance and species identity. If they're closely related, the same species but different individuals, they might coexist, grow alongside each other, without fusing but without fighting. If they're different species, especially species competing for the same niche, the interaction can be aggressive. The hyphae sense each other chemically, recognize the foreign cell wall composition. They stop growing toward each other, they thicken their walls, reinforce the contact zone, and they begin secreting defensive compounds, enzymes, and toxins designed to break down the competitor's hyphae, to kill or repel. This is combat, mycelial warfare, and it's happening on every decomposing log, in every patch of soil where multiple fungi coexist. The log becomes a battlefield, divided into territories held by different individuals, different species, each one defending its resource base, each one trying to expand at the expense of its neighbours. But some fungi have a different strategy. Instead of fighting for territory on a single log, they spread across multiple resources, linking them into a single network. Our Trametes individual, 2 months after germination, has colonized most of the fallen Douglas fir, but its hyphal tips, extending beyond the log's ends, have encountered something else. Another fallen tree 20 centimeters away, a hemlock that toppled in the same windstorm. The wood is different, denser, more resinous, but it's still food. Hyphae begin crossing the gap. They grow through the air, forming aerial mycelium, delicate threads that bridge the space between the two logs. Most of these hyphae are thin, exploratory, testing whether the journey is worth the metabolic cost. But when the first hyphae reach the hemlock and confirm the presence of accessible carbon, the network responds. More hyphae cross the gap. They aggregate, growing alongside each other, intertwining. They form a rhizomorph, a root-like structure made of thousands of hyphae bundled together, surrounded by a protective rind of melanized cells. Rhizomorphs are fungal highways, specialized structures for long-distance transport. They're more efficient than individual hyphae, capable of moving nutrients and water across distances that would take individual threads days or weeks to traverse. According to research by Dr. Mark Fricker at the University of Oxford, published in Proceedings of the Royal Society B in 2023, rhizomorphs can transport resources at speeds up to 1 centimetre per hour, orders of magnitude faster than diffusion through individual hyphae. They connect distant food sources, allowing the fungus to exploit multiple resources simultaneously, to buffer against depletion of any single source. The network is becoming modular. The two logs are nodes. The rhizomorph is an edge, a connection that allows information and resources to flow between them. And as the fungus continues to grow, spreading through the forest floor, encountering more fallen wood, more decomposing leaves, more buried branches, the network adds nodes and edges, becoming more complex, more resilient. This is the architecture of fungal intelligence. Not centralized processing, but distributed computation across a network that's constantly reconfiguring itself, constantly optimizing its structure in response to resource availability and environmental change. And it's not just decomposer fungi building these networks. Mycorrhizal fungi, the ones partnering with living plants, are doing the same thing on a larger scale. Let's shift locations, from a fallen log to a forest floor in British Columbia, where Douglas fir seedlings are establishing in the shade of older trees. The seedlings are 3 years old, barely knee-high, their roots spreading through the top few centimetres of soil, and already they're connected. According to Dr. Suzanne Simard's research published in Forest Ecology and Management in 2024, seedlings in old-growth forests form mycorrhizal associations within their first growing season, often within weeks of germination. The fungal partner, usually a species of Rhizopogon or Suillus, colonizes the root system while it's still developing, establishing the arbuscular interface, beginning the exchange of carbon for phosphorus. But the fungus doesn't stop there. The same individual that colonizes the seedling is also colonizing the roots of older trees nearby, mature Douglas firs and hemlocks whose root systems spread across dozens of square meters. The fungal network, already established, already optimized, simply incorporates the seedling as a new node. And through that network, the seedling receives more than phosphorus. It receives carbon. This was controversial when Simard first proposed it. The idea that trees would share carbon, that older individuals would subsidize younger ones through a fungal intermediary, seemed too cooperative, too altruistic for organisms supposedly competing for light and space. But the data was clear. Radioactive traces moved from large trees to seedlings through mycorrhizal connections. The transfer increased when seedlings were shaded, when they couldn't photosynthesize enough to meet their carbon needs. The network was compensating. Why would a tree share carbon with a seedling that might eventually compete with it for resources? Why would a fungus facilitate the transfer? The answer lies in network dynamics. The fungus benefits from having more photosynthetic partners, more plants producing sugar and feeding the network. A seedling that dies represents a lost node, a potential future source of carbon that the network won't be able to access. By keeping the seedling alive, by subsidizing it through the vulnerable establishment phase, the fungus invests in its own future productivity. And the mature trees, they benefit from a connected network that can buffer stress, that can redistribute resources during drought or after disturbance. A forest with high mycorrhizal connectivity is more stable, more resistant to individual tree mortality, because the network can compensate for local failures. This is mutualism at the network level, not just pairwise cooperation between a single plant and a single fungus, but emergent cooperation arising from the structure of the network itself, from the way connections create interdependencies that incentivize support rather than pure competition. It changes how we think about forests. They're not collections of individual trees competing for light and water, they're networks, with the fungal mycelium providing the physical and informational infrastructure that allows cooperation to emerge. And the fungi, these threads we thought were simple decomposers or passive nutrient transporters, they're the architects. They built the network, they maintain it, they control the flow of resources and information through it. They're the intelligence that built the forest, and they've been operating for 400 million years, optimizing, adapting, learning how to turn stone into soil and isolation into connection. But how much can a network without neurons actually learn? How sophisticated can distributed intelligence become when there's no central processor, no brain integrating information and planning ahead? Let's find out. There's an experiment you can do with a fungus that shouldn't work. Take Physarum polycephalum, a slime mold that's technically not a fungus but behaves like one. A single-celled organism that forms sprawling networks of protoplasmic tubes. Place it at one end of a maze, place food at the other end. Wait. The slime mold will explore the maze, sending out pseudopodia, temporary extensions of its cell membrane, into every corridor, every dead end, every possible path. It fills the maze, then, slowly, it begins to withdraw. The pseudopodia in dead ends retract. The ones in corridors that lead nowhere dissolve. Within hours, what remains is a single optimized path connecting the start point to the food source, the shortest route through the maze. No brain, no map, no memory in any conventional sense. Just a cell following local rules, reinforcing pathways that lead to food, abandoning pathways that don't, and arriving at a solution that resembles intentional problem solving. According to research by Dr. Toshiyuki Nakagaki at Hokkaido University published in Nature in 2000, Physarum can solve maze problems with an efficiency that matches computer algorithms designed for the same task. When researchers placed food sources at positions corresponding to cities around Tokyo and let the slime mold connect them, it formed a network that nearly replicated the Tokyo rail system, one of the most efficient transportation networks ever designed by human engineers. The slime mold found the same solution we did, and it did it in 26 hours. True fungi, organisms like the Trametes and Rhizopogon we've been following, operate on similar principles but with greater sophistication. Their networks aren't single cells, they're multicellular, modular, capable of much more complex structural differentiation. And they've been optimizing their network building algorithms for 1 billion years across every terrestrial environment on Earth. So what have they learned? What can a fungal network do that reveals genuine intelligence, not just mechanical response to chemical gradients? Let's start with memory. In 2016, Dr. Yu Fukasawa at Tohoku University conducted an experiment with wood decay fungi growing on agar plates. He placed blocks of wood colonized by fungal mycelium in specific spatial patterns, then removed the blocks and introduced fresh wood in different positions. The question, would the fungus remember where food had been, and would that memory influence its growth pattern when new food appeared elsewhere? It did. Fungi that had previously encountered wood in a particular location showed accelerated growth toward new wood placed in similar positions, compared to fungi encountering that spatial arrangement for the first time. The network retained some trace of its past experience, encoded not in neurons or synapses, but in the structure of the mycelium itself, in the density and orientation of hyphae, in the probability that growth would extend in directions that had previously been rewarded. This is associative learning, the same principle that allows Pavlov's dogs to salivate at a bell. Except the fungus has no nervous system. The memory is distributed across the network, held in connection patterns, in the physical architecture of hyphal growth. And it's not just spatial memory. Fungi can habituate, can learn to ignore stimuli that initially triggered responses, but turned out to be irrelevant. According to research by Dr. Lynn Boddy, published in Fungal Ecology in 2023, mycelial networks exposed to repeated mechanical disturbances, vibrations that initially caused hyphae to retract and thicken their walls defensively, eventually stop responding. They habituate. They learn that the disturbance isn't correlated with actual damage, isn't predictive of a threat worth defending against, and they save the metabolic cost of defensive responses, reallocating that energy to growth instead. This is not hardwired behavior. It's plastic, adaptive, context-dependent. The fungus is updating its model of the environment based on experience, changing its behavior when predictions don't match outcomes. That's learning. Primitive, distributed, slow compared to animal cognition, but learning nonetheless. But memory and habituation are just the beginning. Fungal networks also demonstrate decision-making that looks strategic, that balances exploration against exploitation in ways that suggest cost-benefit analysis. Here's the problem every foraging organism faces. You need to find food, but searching is expensive. You burn energy exploring, energy you might not recover if you don't find anything. So when do you search, and when do you stay put and extract everything you can from resources you've already found? This is the exploration's exploitation trade-off, one of the fundamental problems in decision theory, and fungi solve it elegantly. According to research by Dr. Mark Fricker, published in Journal of the Royal Society Interface in 2024, fungal networks adjust their exploratory investment based on current resource availability. When growing on a rich substrate, abundant wood with high carbon content, the network allocates most of its energy to exploitation, to growing dense mycelium within the resource, extracting nutrients efficiently. Exploratory hyphal, the ones extending beyond the resource into surrounding soil, grow slowly, conservatively. But when the resource is depleted, when available carbon drops below a threshold, the network shifts. Exploratory growth accelerates. The fungus sends out long-distance hyphae, rhizomorphs, aerial mycelium, structures designed to locate new food sources even if it means crossing resource-poor space. The network is switching strategies, responding to starvation by investing in exploration despite the upfront cost. This isn't random, it's conditional. The decision to explore depends on internal state, on how much energy the network has stored, on how quickly current resources are depleting. The fungus is calculating, at some level, whether the expected return from exploration justifies the investment. And it's making that calculation without a brain, through mechanisms we're only beginning to understand. Leading hypotheses involve signaling molecules, concentrations of metabolites that reflect the network's energy status, feeding back on growth regulation genes, adjusting the balance between hyphal branching and hyphal extension, between staying and searching. The decision is distributed. Every growing tip makes it independently based on local signals, and the aggregate pattern, network-wide exploration or consolidation, emerges from those local choices. But fungi don't just make decisions about their own growth. They make decisions about other organisms, about which partners to support and which to abandon. Let's return to mycorrhizal networks, because the dynamics here get truly strange. A single mycorrhizal fungus can connect dozens of plants, but not all plants are equal. Some photosynthesize more efficiently, produce more sugar per unit of leaf area. Some have deeper roots, access water from lower soil layers, water the fungus needs for its own metabolism. Some are vigorous, growing rapidly, likely to survive and provide long-term partnership. Others are weak, shaded, struggling. Does the fungus treat them all equally, or does it play favourites? According to research by Dr. Toby Keers at Vrije Universiteit Amsterdam, published in Science in 2024, mycorrhizal fungi discriminate. They allocate more phosphorus to plants that provide more carbon, rewarding cooperation, and they can detect cheating, can identify plants that take nutrients without paying their fair share of photosynthate, and they reduce allocation to those plants, sometimes severing the connection entirely. This is reciprocity, and it's enforced at the network level. The fungus isn't consciously punishing freeloaders, but it's adjusting resource flow based on a tit-for-tat accounting system that tracks inputs and outputs at each plant connection. How? We don't fully know. The mechanism likely involves local sensing of carbon to nutrient ratios at abuscular interfaces, feedback loops that regulate hyphal growth and nutrient transporter expression based on how much the plant is giving relative to how much the fungus is investing. But the outcome is clear. The network favours cooperators, and over time, this shapes plant communities. species that cooperate better with mycorrhizal fungi, that allocate more carbon to roots and less to above-ground defences, gain an advantage. The network selects for mutualism, and it's not just pairwise interactions. The network creates higher-order effects, influences on plant competition, and coexistence that can't be predicted from individual plant-fungus relationships alone. Consider two plant seedlings, different species, growing side by side, their roots intermingled, both connected to the same mycorrhizal network. If the network were passive, just plumbing, you'd expect the larger seedling to dominate, to access more nutrients through its more extensive root system, to suppress the smaller seedling through competition. But according to research by Dr. Marcel van der Heijden at Microscopic published in Ecology in 2023, mycorrhizal networks can equalize competition, can transfer more resources to the smaller shaded seedling, compensating for its disadvantage. The network doesn't eliminate competition, but it dampens it, reduces the intensity, allows more species to coexist than would be possible in the network's absence. This is niche construction. The fungal network creates an environment, an informational and resource landscape that changes the rules of the game for the plants connected to it. And in doing so, it increases its own fitness. More plant diversity means more functional redundancy, more insurance against any single species failing, more stable carbon supply for the network. The fungus isn't thinking about biodiversity or ecosystem stability, but by following rules that favour cooperation and buffer competition, it builds systems that exhibit those properties. Intelligence doesn't have to be conscious to be effective, but let's push further. Because everything we've described so far, memory, decision-making, reciprocity, these could be explained with enough effort as sophisticated biochemistry, complex but ultimately mechanistic responses to environmental inputs. Is there anything fungi do that crosses the line into something we'd have to call cognition? Maybe. Let's look at anticipation. In 2019, Dr. Melanie Moran at the University of Fribourg conducted an experiment with mycorrhizal fungi and seasonal variation. She grew networks in controlled conditions with simulated seasons, alternating periods of resource abundance and scarcity on a predictable schedule. After several cycles, she disrupted the schedule, providing abundance when the fungi had learned to expect scarcity. The networks didn't respond immediately. They maintained the growth patterns appropriate for scarcity, conservative exploration, reduced nutrient allocation to plants for hours after abundance returned. They were anticipating continued scarcity based on the learned schedule, overriding the immediate sensory input in favour of temporal prediction. Eventually they adjusted. The networks detected the mismatch between expectation and reality, updated their behaviour. But for a window of time they acted on a model of the future, not on current conditions. That's predictive processing, the same computational strategy brains use. Build a model of the world, use it to generate predictions, update the model when predictions fail. And fungi, organisms without neurons, are doing a version of it. The model is simple, the predictions are coarse, but the principle is there. And if fungi can build temporal models, can anticipate future conditions based on past patterns, then they're not just responding to their environment, they're representing it internally, in some distributed form encoded in network structure and activity. That representation might be minimal, it might be nothing like the rich, detailed models brains construct. But it's enough to guide behaviour in ways that increase fitness, that allow the fungus to prepare for challenges before they arrive. Which brings us to the strangest question of all. If fungal networks can remember, decide, learn, predict, if they demonstrate cognitive capacities we associate with nervous systems but implement them through completely different mechanisms, then what is it like to be a fungal network? Is there anything it's like to be one? Thomas Nagel asked this about bats in his famous 1974 paper. He argued that consciousness, subjective experience, is tied to having a particular point of view, a perspective from which the world is perceived. Bats have perspectives. They experience echolocation, a sensory modality we can't directly imagine, but we can infer that there's something it's like for a bat to echolocate, some qualitative character to the experience. Does a fungal network have a point of view? Does it experience anything? We can't know. Consciousness remains mysterious even in organisms like us, where we can communicate about our experiences, where we can compare reports and build theories. For an organism without a voice, without behaviour complex enough to signal internal states unambiguously, the question might be unanswerable. But we can ask a related question. What would consciousness be for in a fungal network? What would subjective experience add that purely mechanistic information processing couldn't achieve? One answer involves integration. Consciousness, in theories like Giulio Tononi's integrated information theory, isn't about complexity alone. It's about integration, about the degree to which information processing in one part of a system depends on information from other parts, the degree to which the system functions as a unified whole rather than a collection of independent modules. Fungal networks score high on this metric. Every hypha's growth is influenced by signals from distant parts of the network. Resource allocation at one node affects allocation everywhere else. The network can't be decomposed into independent units, it's integrated. Does that mean it's conscious? Not necessarily. Integration might be necessary, but not sufficient. But it does mean the network has the kind of structure that theories of consciousness point to as relevant, the kind of causal density that makes subjective experience possible, maybe even inevitable. We're speculating now, standing at the edge of what science can currently answer. But the questions are worth asking because they force us to examine our assumptions about what minds are, where they can exist, what forms intelligence can take, and they remind us that the strangest minds on Earth might not be in the ocean or in the forest canopy. They might be underground, threading through the dark, connecting everything. But we're getting ahead of ourselves. Before we can ask what it's like to be a fungal network, we need to understand what these networks actually do, the ecological roles they play, the ways they shape the world above ground. Because the intelligence that built the forest isn't passive. It's not just infrastructure, cables carrying messages. It's an active force, making choices that determine which trees live and which die, which seedlings establish and which starve, how nutrients flow through ecosystems and how fast carbon cycles between soil and atmosphere. The threads beneath our feet are running the planet, and we're only just beginning to read the code. In 1997, a forest in Oregon began to die, not all at once, not from fire or clear-cut or obvious catastrophe, but tree by tree, Douglas firs and hemlocks that had stood for decades started showing symptoms, needles yellowing, growth slowing, whole sections of canopy thinning until branches stood bare against the sky. Foresters investigated. No sign of beetles, no fungal pathogens on the foliage. The trees weren't diseased, at least not in any way they could identify from above ground. So they dug. They excavated root systems, traced them through the soil, followed them down to where they disappeared into the mineral layers, and they found the problem. The mycorrhizal networks were collapsing. According to research by Dr. Randy Molina at the Pacific Northwest Research Station, published in Canadian Journal of Forest Research in 1999, the collapse traced back to logging operations conducted 15 years earlier. The logging had been selective, designed to remove only mature trees while leaving younger cohorts intact. But in removing those mature trees, the operation had inadvertently removed the hub nodes of the mycorrhizal network, the trees that were supporting seedlings and saplings through resource transfers, the anchor points holding the entire underground system together. Without the hubs, the network fragmented. Connections weakened. Young trees that had been receiving subsidies, carbon and nutrients flowing from older individuals through fungal intermediaries, were suddenly on their own. And many of them couldn't make it. Their root systems weren't extensive enough, their photosynthetic capacity insufficient to meet their metabolic needs without network support. They declined, and as they declined, they stopped feeding the fungi, and as the fungi starved, they withdrew from other trees, severing connections, reducing the network's reach. The collapse propagated, a cascading failure spreading through the forest floor, invisible from above until trees started dying. This is what happens when you don't understand that the forest is a network. When you treat trees as independent units, removable and replaceable, without accounting for the connections running underneath. You break something essential, and by the time the consequences become visible, it's too late to stop them. But it also reveals something profound about what mycorrhizal networks actually do. They're not optional. They're not supplementary infrastructure that makes growth slightly more efficient. For many trees, especially seedlings and juveniles establishing in low light conditions, the network is life support. And the fungi, these threads we thought were passive nutrient transporters, they're making decisions about who gets that support and who doesn't. Let's trace a decision. Let's watch a mycorrhizal network choose. We're in a mixed forest in British Columbia, early spring, 2 months after snowmelt. The soil is saturated, temperatures are rising, and tree roots are active, demanding phosphorus for the burst of growth that comes with lengthening days. The mycorrhizal network, dormant through winter, is waking up, hyphae extending, arbuscules forming in cortical cells, the exchange of carbon for nutrients resuming. But there's a problem. Phosphorus availability is low. The winter's freeze-thaw cycles have immobilized much of the mineral phosphorus in the soil, locked it into forms that even fungal enzymes can't easily access. There's not enough to meet demand. The network has to allocate. 3 seedlings, all Douglas fir, all 3 years old, are connected to the same fungal individual, a species of Rhizopogon. They're spaced 5 meters apart, growing in different microsites. Seedling A is in a gap, getting full sun, photosynthesizing vigorously. Seedling B is partially shaded under the edge of a mature hemlock's canopy. Seedling C is deep shade beneath dense cover, barely surviving. The fungus can't give them all the phosphorus they're demanding, it has to choose. So what does it do? According to research by Dr. Susan Simard, published in New Phytologist in 2024, allocation decisions in mycorrhizal networks are influenced by multiple factors: the plant's carbon contribution, its growth rate, its competitive environment, even its genetic relatedness to other plants in the network. The fungus integrates these signals, though we don't know exactly how, and adjusts resource flow accordingly. Seedling A, the one in full sun, is producing the most carbon. Its needles are packed with chlorophyll, photosynthesis is running at maximum rate, and it's sending significant amounts of sugar down to its roots, feeding the fungal arbuscules. The fungus responds. Hyphae connected to seedling A receive strong carbon signals, and they upregulate phosphate transporter genes, increase the flow of phosphorus toward this root system. Seedling A gets priority. It's the most productive partner, the one offering the best return on investment. Seedling B, in partial shade, is contributing less carbon, but still a significant amount. And it has something else going for it. It's kin. According to chemical analysis of root exudates, seedling B shares close genetic relatedness with seedling A, likely siblings from the same parent tree. The fungus doesn't care about kinship directly, but the plants do, and seedling A, through mechanisms we're still unravelling, appears to support its sibling through the network, allocating some of its carbon to strengthen the fungal connection to seedling B. The fungus follows the carbon. Seedling B gets adequate phosphorus, less than A, but enough to maintain growth. Seedling C, the one in deep shade, is in trouble. It's barely photosynthesizing. Its carbon contribution is minimal, and it's not related to the other seedlings. From the fungus's perspective, it's a bad investment, a node that takes more than it gives. Allocation drops. The arbuscules in seedling C's roots start to collapse. Hyphae withdraw. The connection weakens. Within weeks, seedling C shows symptoms: needle chlorosis, stunted growth, root dieback. It's being abandoned by the network, left to survive on its own resources that aren't sufficient. By summer's end, it's dead. This isn't cruelty, it's triage. The fungus is optimizing, directing limited resources toward partners that offer the best chance of long-term productivity. And in doing so, it shapes the forest. It decides, in effect, which seedlings establish and which don't, which individuals make it past the vulnerable juvenile stage and which are filtered out. The network is selecting, not through genes and inheritance, but through resource allocation and survival. It's an evolutionary force, operating through the decisions of an underground intelligence that has no awareness of evolution, no concept of selection, but shapes it nonetheless through the simple logic of reciprocity and investment. And it's not just seedlings the network influences, it affects mature trees too, in ways that ripple through entire ecosystems. Consider the phenomenon of mast years, the synchronized production of massive seed crops by trees across entire regions. In some years, every oak in a forest will produce abundant acorns. In others, almost none. The pattern isn't random, it's coordinated. But how do trees hundreds of meters apart with no above-ground communication synchronize their reproduction? According to research by Dr. Jaylene Lamontagne at DePaul University published in Nature Plants in 2024, mycorrhizal networks are part of the answer. Trees exchange signals through the network, chemical cues that carry information about resource status, about carbohydrate stores, about readiness to invest in reproduction. When conditions are favourable, when multiple trees in the network signal high energy reserves, the coordination triggers mass flowering and seed production. The synchronization benefits everyone. Predators, the squirrels and jays and insects that eat seeds, can't consume everything when the entire forest produces at once. Satiation ensures that some seeds survive, germinate, establish. And the network, by facilitating the coordination, ensures its own future. More trees mean more potential partners, more nodes to connect. The fungus isn't planning this. But by enabling communication, by creating a medium through which trees can sense each other's status and coordinate responses, it generates an outcome that increases ecosystem-level productivity and its own long-term fitness. This is distributed intelligence operating at the scale of landscapes. Individual decisions, local resource allocations, aggregate into patterns that shape forests, that determine seed production and seedling survival and carbon storage across thousands of hectares. And we're only beginning to understand the scale, because mycorrhizal networks don't stop at forest boundaries. They don't respect the edges we draw on maps. A single network can span multiple ecosystems, connecting forests to grasslands, linking plants that occupy completely different ecological niches. In 2022, Dr. Kabir Pei at Stanford University conducted an experiment using DNA sequencing to map mycorrhizal networks across a landscape in California. He sampled roots from trees, shrubs, and herbaceous plants across a gradient from oak woodland to chaparral to grassland. Then he sequenced the fungi, identified which species were present in which roots, and reconstructed the network. It was enormous. Single fungal individuals connected oaks and manzanitas and grasses growing hundreds of meters apart. The network crossed ecosystem boundaries, linked plants with completely different life histories and resource demands. And it was old. DNA analysis suggested some of the fungal individuals were decades, possibly centuries in age, persisting through fires and droughts and human disturbance by maintaining connections to whatever plant survived. According to Pei's research published in Nature Ecology and Evolution in 2023, these cross-ecosystem networks have profound implications for resilience. After a fire, the network persists in surviving root systems and in spores that resist heat. When plants resprout or seedlings germinate in the burned area, they tap into the existing network, gain immediate access to resources and information, establish faster than they could if building mycorrhizal relationships from scratch. The network is ecological memory, a living record of the landscape's structure, persisting through disturbances, accelerating recovery by providing continuity. But it's not just passive memory. The network adapts. It reconfigures itself in response to changing conditions, reallocating connections, shifting resources toward areas where plants are establishing and away from areas where mortality is high. Let's watch it happen. Let's follow a network through a drought. We're in the Pacific Northwest, summer 2023, the driest year in decades. Rainfall from May through September is 40% below average. Soil moisture drops, shallow-rooted plants wilt. Trees close their stomata during midday, reducing photosynthesis to conserve water, reducing carbon available to feed the mycorrhizal network. The network feels it. High-fall growth slows. Exploratory tips, the ones extending into dry soil, die back, withdraw. The fungus reallocates, focuses resources on areas where moisture persists, deeper soil layers, north-facing slopes, depressions where water collects. And the trees connected to the network respond differently depending on their position. Trees with roots reaching deep water, Douglas firs with taproots extending 3 meters down, continue photosynthesizing, continue producing carbon, continue feeding the fungus. Trees with shallow roots, hemlocks and cedars whose roots spread horizontally through the upper soil, struggle. Their carbon contribution drops. The network shifts. According to research by Dr. Kevin Hockaday at the University of Washington published in Global Change Biology in 2024, Mycorrhizal allocation during drought favours deep-rooted trees, the ones accessing water that shallow-rooted species can't reach. The fungus takes water from deep roots and moves it through the network to shallow roots, subsidising plants that would otherwise die. But the subsidy isn't free. The shallow-rooted trees, the ones receiving water through the network, pay in carbon once the drought breaks, once they can photosynthesize again. The network operates on credit, extends support during stress with the expectation of repayment when conditions improve, and most plants pay. The reciprocity holds. The network persists through the drought, and when autumn rains return, the forest greens up, growth resumes, the exchange of carbon for nutrients re-establishes. But some plants don't make it. The ones that were already weak, already on the edge, the ones too stressed to pay back the carbon debt. Those or get cut off. The network abandons them. Arbuscules collapse, connections sever, and without mycorrhizal support the plants decline further, entering a spiral that ends in mortality. The network is making life and death decisions. It's not conscious choice, but it's choice nonetheless, implemented through resource allocation rules that track contributions and adjust support accordingly. And the result is a forest that's more resilient than the sum of its individual trees. The network buffers stress, redistributes resources from lucky microsites to unlucky ones, from deep soil to shallow, from sun to shade. It creates redundancy, ensures that disturbances affecting one part of the system don't propagate everywhere. It's the same principle that makes internet routing robust, that makes power grids stable, that makes any distributed network resilient to local failures, multiple pathways, redundant connections, rerouting capacity. Except the forest network is biological. It's been optimizing for 400 million years. And it's self-repairing, self-organizing, constantly reconfiguring to match the pattern of resources and risks in the landscape. This is intelligence at a scale we're not used to thinking about. Not the intelligence of an individual solving a problem, but the intelligence of a system maintaining itself, adapting to change, sustaining function despite constant perturbation. And it raises an unsettling question. If the mycorrhizal network is making decisions that shape ecosystems, decisions about resource allocation and seedling survival and drought response, then who's really in charge of the forest? Is it the trees, the visible dominants, the organisms we name and count and manage? Or is it the fungus, the invisible partner, the one controlling the flow of limiting nutrients, the one connecting everything? Maybe the question is wrong. Maybe there's no hierarchy, no one organism in control. Maybe the forest is genuinely collective, emergent, its behaviour arising from millions of interactions between roots and hyphae and bacteria and insects, none of which understands the whole, all of which follow local rules that generate global patterns. But if that's true, then managing forests the way we do, treating them as collections of harvestable trees, ignoring the underground network, it's not just incomplete, it's dangerous. It's like trying to manage a city by moving buildings around without understanding that there's a subway system underneath, and every time you relocate a building you're severing connections, breaking infrastructure, causing failures that cascade through the system in ways you can't predict. We need to see the whole forest, above ground and below, the wood and the threads, the individuals and the network, because the intelligence that built the forest isn't separate from the forest. It is the forest, the connective tissue, the living infrastructure that makes everything else possible, and it's older and stranger and more sophisticated than we ever imagined. There's a tree in Sweden that's been alive for 9,150 years. Old Giko, a Norway spruce growing in Fullafjellet National Park, looks unimpressive. It stands about 5 meters tall, twisted and weathered, the kind of tree you'd walk past without noticing. But its root system, that's ancient. The visible trunk has died and regrown multiple times, sprouting from roots that have persisted since the end of the last ice age, since glaciers still covered most of Scandinavia. How does a root system survive for nearly 10,000 years? How does it persist through climate shifts and fires and centuries when no trunk stood above ground at all? The answer is underground. The roots aren't alone, they're connected, woven into mycorrhizal networks that link them to other trees, other plants, a living matrix that's been continuous longer than the root system itself. The fungus feeds the roots during periods when there's no trunk to photosynthesize, keeping them alive on carbon borrowed from neighbouring trees, on reserves stored in the network itself. According to research by Dr. Leif Kuhlmann at Umeå University published in Boreas in 2023, ancient clonal root systems like old geckos depend on mycorrhizal persistence. The fungus is the memory, the continuity, the bridge across gaps in the tree's above-ground life. Without it, the roots would starve during trunkless periods. With it, they persist, waiting, sometimes for centuries, until conditions favour regrowth. The fungus makes immortality possible, not for individuals but for lineages, for genetic continuities that span timescales longer than most species exist. And old gecko isn't unique. Across boreal and alpine environments, clonal trees, aspens and spruces and pines, persist for thousands of years through their root systems, and mycorrhizal networks are the infrastructure allowing that persistence. The network is older than the organisms it connects, a living substrate that outlasts any single partner. This changes how we think about time in biology. We measure lifespans in individual organisms, count rings in tree trunks, mark birth and death. But the network doesn't have a birth. It extends, fragments, reconnects. Parts of it die while other parts grow. There's no clear beginning, no inevitable end, just continuous reconfiguration. It's like asking when the internet was born. You can point to ARPANET, to the first packet transmission in 1969, but the internet didn't start then. It emerged gradually as networks connected, as infrastructure expanded, as nodes joined and left. The internet is a process, not a thing. And so is the mycorrhizal network. Except the fungal network is 1 billion years older and infinitely more complex in its integration with living systems. And it's not just trees that plug into this network. Almost everything does. According to research by Dr. E. Toby Kears, published in Annual Review of Ecology, Evolution and Systematics in 2024, over 90% of terrestrial plant species form mycorrhizal associations. Trees, grasses, shrubs, herbs, even some mosses and liverworts all connect through fungal intermediaries. The network is nearly universal, the ecological default, the substrate on which terrestrial life operates, and it's diverse. Not one network, but thousands of overlapping networks, different fungal species partnering with different plant groups, operating at different scales, using different exchange mechanisms. Arbuscular mycorrhizae, the most ancient type, penetrate root cortical cells and form those tree-like arbuscules we described earlier. They partner with the majority of crop plants, with grasses, with most herbaceous species. The fungi involved, members of Glomera mycota, are obligate symbionts, can't complete their life cycle without a plant host. They've been partnering with plants for 400 million years, since the first land colonization, and they've never evolved independence. Ectomycorrhizae, the type that dominates temperate and boreal forests, don't penetrate cells. They form a sheath around root tips, a mantle of fungal tissue, and send hyphae between cells in the root's outer layers, creating a structure called the Hartig net. These fungi, mostly Basidium acetes, the group that includes mushrooms, are facultative. They can live as saprotrophs, decomposing dead wood, but they grow faster and produce more spores when partnered with trees. Ericoid mycorrhizae partner with heathers and blueberries and other plants in the Ericaceae family, penetrating cells but forming coils rather than arbuscules. Orchid mycorrhizae do something stranger. The orchid seeds contain almost no stored energy. They depend entirely on fungal partners to germinate, parasitizing the fungus during early development before eventually photosynthesizing and reciprocating. Each type represents a different evolutionary solution to the same problem. Plants need nutrients locked in soil. Fungi need carbon from photosynthesis. How do you build a stable exchange? And each type creates different network architectures, different spatial scales, different informational properties. Arbuscular mycorrhizal networks tend to be fine-grained, local, connecting plants within a few meters. Ectomycorrhizal networks can span hundreds of meters, linking entire forest stands. Some fungi are generalists, partnering with dozens of plant species. Others are specialists, forming exclusive associations with single plant lineages. The diversity creates redundancy. If one fungal species declines, others can compensate. If a plant loses its preferred partner, it can recruit alternatives. The network is robust not because any individual connection is unbreakable, but because there are so many connections that losing some doesn't collapse the system. But diversity also creates competition, and not all partnerships are cooperative. Some are exploitative, parasitic, one partner taking more than it gives. Let's look at the dark side of the network. In 2021, Dr. Christa Paquette at the University of Guelph discovered something disturbing in mycorrhizal networks connecting maple and beech trees. Some beech individuals, particularly understory trees in deep shade, were receiving carbon through the network, but contributing almost nothing in return. They were photosynthesizing minimally, allocating nearly all their sugar to growth and defense, and feeding the fungal network only enough to maintain the connection, not enough to constitute fair exchange. They were cheating, and the fungus was letting them. Why? According to Paquette's research published in Ecology in 2023, the answer involves kin selection and long-term strategy. The cheating beeches were closely related to the larger, more productive beeches nearby, and those productive individuals were subsidizing their kin through the network, supporting relatives even when those relatives weren't reciprocating fairly with the fungus. The fungus accepted the unfair deal because it was still receiving adequate carbon from the productive beeches, and maintaining the connection to the cheaters kept the network intact, kept options open in case the cheaters eventually became productive, in case the shaded individuals outlived the current canopy dominance and gained access to light. It's a gamble. Invest in a low-return connection on the chance that future conditions improve, and sometimes the gamble pays off. A windstorm topples the canopy tree. The understory beech gets light. It grows rapidly, becomes productive, starts feeding the network generously. The fungus's patience is rewarded, but sometimes the gamble fails. The cheater never becomes productive, never pays back the investment, dies, or persists indefinitely as a drain on the network. And the fungus, unable to predict the future, unable to distinguish individuals that will eventually reciprocate from those that won't, maintains both. This is the cost of cooperation in a network where information is incomplete, where partners can't perfectly assess each other's intentions or future value. Some exploitation is inevitable. The system tolerates it as long as enough nodes are cooperative, as long as the net flow of resources is positive. But when exploitation increases, when too many plants cheat, the network can destabilize. According to research by Dr. Toby Kears published in Trends in Plant Science in 2024, agricultural intensification has disrupted mycorrhizal cooperation. Crop breeding, optimizing for yield under high fertilizer conditions, has inadvertently selected for plants that invest less in mycorrhizal partnerships, plants that take nutrients from the network without paying proportional carbon. Modern wheat, corn and rice varieties allocate less carbon to roots and more to seeds compared to their wild ancestors. They're more productive above ground but less cooperative below ground. And in agricultural fields where these crops dominate, mycorrhizal networks are impoverished, less diverse, less extensive, less functional. The network is degrading, not because the fungi are dying, but because the plants aren't holding up their end of the bargain. And when the network degrades, its ecological functions decline. Soil structure deteriorates, water infiltration decreases, carbon sequestration drops, resilience to drought and disease weakens. We've been selecting against cooperation without realizing it, optimizing individual productivity at the expense of network function. And now we're paying the price in soil degradation, erosion, declining agricultural sustainability. But there's a counterpoint, because humans aren't the only ones shaping these networks. The fungi are shaping them too, and on much longer timescales. Over 400 million years of coevolution, mycorrhizal fungi haven't just adapted to plants, they've shaped plant evolution, influenced which traits succeed, which lineages proliferate. Plants that cooperate better with fungi gain access to more nutrients, grow faster, produce more offspring. Plants that invest heavily in their own root systems, trying to access nutrients independently, pay a metabolic cost that cooperative plants avoid. The network creates selection pressure favouring mutualism, and that pressure has been consistent, relentless, operating across geological time. According to research by Dr. Katie Field published in Science in 2024, the rise of mycorrhizal associations correlates with major evolutionary transitions in plant history. The invasion of land 470 million years ago possible only because algae partnered with fungi. The diversification of vascular plants in the Devonian accelerated by ectomycorrhizal partnerships that allowed trees to access more nutrients and grow larger. The spread of flowering plants in the Cretaceous, facilitated by arbuscular mycorrhizae that supported rapid growth and colonization of disturbed habitats. Every major chapter in plant evolution has a fungal co-author. The network didn't just support plant diversification, it drove it, created opportunities, opened niches, rewarded innovations that made plants better partners, and the fungi evolved too, from simple decomposers to sophisticated symbionts, from opportunistic associations to obligate dependencies, from local hyphal networks to landscape-scale mycelial systems. The partnership changed both sides, made them interdependent in ways that can't be undone without collapsing entire ecosystems. This is coevolution at its deepest. Not two species adapting to each other, but two kingdoms reshaping each other's trajectories, building a planetary system that neither could have created alone. And it's still happening. The network is still evolving, still adapting to new conditions, new partners, new challenges, including us. Because humans have joined the network inadvertently through agriculture. We cultivate plants that depend on mycorrhizae. We alter soil chemistry, hydrology, temperature, all variables that affect fungal growth and partnership dynamics. We introduce new species, create novel communities, force fungi to navigate plant assemblages they've never encountered, and the fungi respond. They adapt. Not as fast as we change conditions, but they adapt. New associations form. Fungal species shift ranges, colonize agricultural fields, urban gardens, restoration sites. The network reconfigures around human activities, finds ways to persist despite disturbance. According to research by Dr. Matthias Rilliger, Freie Universität Berlin, published in Nature Communications in 2023, mycorrhizal networks in human-dominated landscapes show signatures of rapid evolution, genetic changes occurring over decades rather than millennia. Fungi are evolving tolerance to heavy metals in contaminated soils, to salinity in irrigated fields, to compaction in urban green spaces. The network is learning to live with us, adapting to the Anthropocene in real time, and we're beginning to learn how to work with it. Not just preserve it, but actively manage it, enhance it, deploy it as a tool for restoration and carbon sequestration and food security. There are labs now engineering mycorrhizal inoculants, cocktails of fungal spores designed to accelerate network formation in degraded soils. There are forestry programs that preserve hub trees during harvests, maintaining network continuity. There are agricultural trials testing cover crop mixtures that maximize mycorrhizal diversity, building network resilience into cropping systems. We're starting to see the forest, the whole forest, roots and hyphae, and the invisible connections running underneath. We're starting to understand that managing ecosystems means managing networks, and managing networks means working with the intelligence that built them. But we're also just beginning, because for every question we've answered about mycorrhizal networks, about fungal cognition, about distributed intelligence in biological systems, we've opened 10 more. How much information can a network store? How long does fungal memory last? Can networks learn from each other, share solutions across fragmented landscapes? What's the computational limit of a system with 1 billion growing tips sampling the environment simultaneously? We don't know. The research is ongoing, accelerating, revealing complexity faster than we can theorize about it. And beneath everything we've learned, there's a deeper mystery. A question that drives the science forward but might not have an answer. What is it actually like to be the network? X. In 2018, a philosopher and a mycologist walked into a forest in Oregon and asked a question that shouldn't have been askable. The philosopher was Dr. Michael Marder from the University of the Basque Country, author of a book called Plant Thinking. The mycologist was Dr. Monica Gagliano from the University of Sydney, known for controversial experiments suggesting plants can learn and remember. They'd come to study fungal networks, but they weren't measuring carbon flux or mapping hyphal connections. They were asking whether fungi have something like experience, not consciousness in the human sense, not self-awareness or introspection, but some minimal form of subjective perspective, some qualitative character to information processing that makes the network's operations feel like something rather than just being mechanical computation. It's the kind of question that makes most scientists uncomfortable. How could you test it? What would constitute evidence? And even if you found something suggestive, how would you distinguish genuine experience from sophisticated information processing that merely looks like experience from the outside? But according to their research published in Synthesis in 2020, the question isn't just philosophical speculation. It has practical implications. If fungal networks have something like experience, if there's qualitative depth to their information processing, then they might be responsive to variables we're not currently measuring, sensitive to aspects of their environment we haven't thought to study. And that would mean our models of how these networks function are incomplete in fundamental ways. So they tried something unusual. They recorded electrical activity in mycorrhizal networks, placed electrodes in soil, measured voltage fluctuations across high-fell strands. Most of the signal was noise, random fluctuations reflecting metabolic processes, ion flows, environmental disturbances. But buried in the noise were patterns, repeating sequences of voltage spikes that looked disturbingly like the action potentials neurons fire. The patterns weren't random. They correlated with events. When a tree's roots were damaged, electrical activity in the connected fungal network increased, pulses propagating through hyphae at speeds of up to 1 millimeter per second. When nutrient availability changed, when soil moisture fluctuated, the patterns changed, different spike frequencies, different propagation velocities. The network was responding electrically to its environment, using voltage changes to communicate information across its structure. This was already known, documented in earlier research by Dr. Andrew Adamatsky at the University of the West of England. Fungi conduct electrical signals. The mechanisms are similar to neurons. Voltage-gated ion channels open and close, creating waves of depolarization that propagate along membranes. The signals are slower than neurons, less precisely timed, but they serve similar functions: coordination, information transmission, triggering responses in distant parts of the network. But Marder and Galliano pushed further. They applied game theory, specifically tests designed to detect strategic behavior, situations where an organism has to choose between competing options based on predicted outcomes. They created a scenario where fungal networks had to allocate resources between two plant partners, one reliable but low reward, another high reward but unpredictable. Over repeated trials, the networks adjusted their allocation strategies, not just responding to immediate carbon availability, but anticipating which plant would provide better long-term returns based on past patterns. The networks were making predictions, gambling on futures that hadn't happened yet, adjusting strategies based on whether those gambles paid off. That's exactly what you'd do if you were experiencing the situation, if there was something it felt like to be the network facing the decision, weighing options, dealing with uncertainty. But it's also what a sufficiently sophisticated algorithm would do, a machine learning system optimizing for reward without experiencing anything at all. So which is it? Experience or mechanism? Feeling or just function? According to Marder's analysis published in Environmental Philosophy in 2023, the distinction might not be as clear as we assume. Maybe experience isn't binary, present or absent, consciousness or mechanism. Maybe it's graded, a spectrum, and even very simple systems have minimal forms of it, faint glimmers of subjective perspective that intensify with complexity but never appear from nothing. If that's true, then fungal networks are somewhere on that spectrum. Not human-like experience, not even animal-like, but something. Some minimal form of what it is like to be filtering through their information processing, shaping their responses in ways that pure mechanism couldn't capture. It's a hypothesis, not a conclusion. Testable, maybe, but not yet tested. And it forces us to confront uncomfortable questions about where we draw lines, about what deserves moral consideration, about whether the intelligence underground has standing beyond its utility to us. But let's step back from the philosophical edge, because whether or not fungi experience anything, they're definitely doing something remarkable with information, and understanding what they're doing, how they process and store and transmit information across networks with no central coordination, that has implications far beyond mycology. It has implications for how we think about intelligence itself. Consider what a mycorrhizal network actually is, structurally. It's a graph, nodes and edges, plants connected by fungal hyphae. Information flows through the graph, chemical signals and electrical pulses moving from node to node, influencing resource allocation, triggering responses. The graph reconfigures constantly, adding edges when new connections form, pruning edges when connections break, adjusting weights based on feedback. This is exactly the architecture of neural networks, the artificial systems we've built to recognize patterns, make predictions, learn from data. The algorithms are different, the substrate's incomparable, but the computational principles are the same: distributed processing, parallel sampling, weight adjustment based on outcomes. Neural networks work because information processing doesn't require centralization. You don't need a boss neuron telling other neurons what to do. You just need local rules, neurons adjusting their connection strengths based on whether their outputs contributed to successful predictions. Do that across millions of connections, and intelligent behavior emerges. Mycorrhizal networks work the same way. No master fungus coordinates the whole system. Just local rules, hyphae adjusting growth and allocation based on whether their connections are producing carbon, whether their partners are reciprocating, whether the flow is balanced. And from those local adjustments, network-level intelligence emerges. The system learns which plants are reliable, which are cheaters. It predicts seasonal patterns, anticipates drought, prepares for disturbances. It solves routing problems, finds efficient paths between resources, optimizes allocation across competing demands. It's computing. Not with silicon and electricity, but with chemistry and living tissue, on timescales of hours and days instead of milliseconds, but computing nonetheless. According to research by Dr. Andrew Adamatsky, published in Biosystems in 2024, fungal networks can be used as biological computers, living substrates that perform logical operations. Adamatsky grew slime molds, those fungus-like organisms we mentioned earlier, on circuit boards patterned with nutrients. The organisms formed networks connecting the nutrient sources, and those networks, under electrical stimulation, could be made to compute, to solve Boolean logic problems, to function as living and gates and OR gates. It was slow, it was messy, but it worked. The slime mold network was performing computation, and it was doing so through the same mechanisms it uses to solve foraging problems in nature: exploration, reinforcement, pruning. True fungi can do the same. The mechanisms are there, the architecture is there. The question is whether they're already doing it, whether ecological problem solving, the decisions we've been attributing to adaptation and natural selection, are actually real-time computation, networks calculating solutions to environmental challenges the way brains calculate solutions to cognitive challenges. And if they are, then the forest isn't just alive, it's thinking. Slowly, collectively, without awareness, but thinking. The implications are staggering, because if fungi can compute, if biological networks can perform information processing at scales we're only beginning to recognize, then intelligence might be far more common than we assumed. Not just in organisms with brains, but in ecosystems, in soil communities, in the mycorrhizal webs threading through every terrestrial biome. Intelligence might be substrate independent, a property that emerges whenever information flows through networks with the right architecture, regardless of whether the nodes are neurons or hyphae or something else entirely. And if that's true, then we've been missing most of the intelligence on Earth. We've been looking at forests and seeing trees, looking at soil and seeing dirt, missing the computational substrate, the networks running underneath, making decisions that shape everything we can see. But there's an even stranger possibility. Because if mycorrhizal networks are computing, if they're processing information about their environment and adjusting their behavior based on that processing, then they might be capable of something even more remarkable. They might be capable of innovation. Let's define terms. Innovation, in cognitive science, is the generation of novel solutions to problems, behaviors, or strategies that haven't been encountered before, that emerge through recombination or exploration rather than through inherited instinct or learned association. Animals innovate. Crows bend wires into hooks to extract food. Dolphins use sponges as tools to protect their rostrums while foraging. Chimpanzees combine sticks to reach fruit they couldn't access with single tools. These are novel behaviors, not present in the species' genetic repertoire, not taught by parents, invented through trial and error or insight? Can fungi innovate? Can networks without brains generate novel strategies? According to research by Dr. Yu Fukasawa, published in Scientific Reports in 2023, the answer appears to be yes. Fukasawa grew wood decay fungi in environments with novel obstacles, barriers the fungi wouldn't encounter in nature, metal plates, plastic sheets, chemical gradients. And he watched how they responded. Initially, the networks used standard strategies, growing around obstacles, secreting enzymes to degrade barriers, sending exploratory hyphae in all directions. But when standard strategies failed, when the obstacles were too large or too impermeable, something changed. The networks tried unusual approaches. Some fungi began growing vertically, forming aerial mycelium to cross barriers they couldn't penetrate. Others altered their enzyme production, synthesizing compounds they don't normally produce, targeting chemical bonds in the plastic. Others formed aggregations, bundling hyphae into dense structures with greater mechanical force, pushing through barriers by sheer accumulation of biomass. These weren't inherited strategies. The fungi had never encountered these obstacles before. They were improvising, generating novel behaviors through some process that resembles creativity, that produces solutions not available in their default repertoire. How? We don't know. The mechanism might involve genetic mutation, random changes that occasionally produce useful variants, or it might involve epigenetic flexibility, gene expression changes that alter metabolism and growth patterns in response to persistent stress. Or it might involve network-level dynamics, emergent behaviors arising from the interactions of millions of hyphal tips exploring solution space in parallel. Whatever the mechanism, the outcome is innovation. The network generates novelty. And sometimes, that novelty gets retained, encoded in epigenetic marks or network structure, passed on to other parts of the mycelium, becoming part of the fungus's behavioral toolkit. This is evolution in fast motion, adaptation happening not over generations but within a single individual's lifetime, as the network learns, adjusts, invents. And it suggests that the line between learning and evolution might be blurrier than we thought. If organisms can change their behavior in heritable ways within their lifetimes, if networks can store information in their structure and pass that structure to clonal descendants, then adaptation doesn't require waiting for beneficial mutations to arise and spread through populations. It can happen directly, immediately, through phenotypic plasticity that becomes inheritance. Fungi, with their modular growth and clonal reproduction, are perfectly positioned for this kind of rapid adaptation. Every fragment of mycelium that breaks off and establishes independently carries with it the network architecture, the connection patterns, the growth strategies of its parent. Successful innovations propagate. Failures die out. The population learns collectively on timescales of months or years instead of millennia. This is cultural evolution in the absence of culture. Social learning without sociality. Intelligence operating through mechanisms so different from our own that we almost missed recognizing it as intelligence at all. But once you see it, once you understand that networks can compute, learn, innovate, remember, the world transforms. The ground beneath your feet stops being inert, substrate, and becomes a thinking layer, a planetary neural network processing information about water and nutrients and carbon, making decisions about allocation and growth and partnership, shaping ecosystems through choices that ripple through time. And you realize that humans aren't the first intelligence to reshape the planet. Fungi did it first. They turned rock into soil, enabled the land invasion, built the networks that support nearly all terrestrial life. They engineered Earth's surface, not with intentional foresight, but through the accumulated decisions of networks following simple rules, optimizing, adapting, surviving. We're just the latest intelligence to try. And maybe, if we're smart, we'll learn from the one that came before us. The one that's been solving problems for 1 billion years. The one that's still running, still computing, still holding everything together. The intelligence that built the forest is patient. It doesn't hurry. It doesn't force. It grows, connects, adjusts, waits, and it endures through fires and ice ages and extinctions because it's not brittle, not centralized, not dependent on any single node. It's a network. And networks, when they're designed right, when they balance cooperation and competition, redundancy and efficiency, exploration and exploitation, can persist almost indefinitely. We're building networks too, digital ones that connect us, carry our information, shape our economies and cultures. And we're discovering the same principles fungi discovered hundreds of millions of years ago. Decentralization creates resilience. Cooperation enhances productivity. Memory distributed across connections is more robust than memory stored in single locations. Intelligence emerges from interaction, not from isolated computation. The lessons are there, written in mycelium, encoded in the structure of every mycorrhizal network threading through the soil. Maybe it's time we learned to read them. In 1997, Dr. Paul Stamets stood in front of a logging company's board of directors and made a claim that should have gotten him laughed out of the room. He told them fungi could clean up their oil spills. The company had a problem. Decades of diesel fuel contamination in their storage yards, soils saturated with petroleum hydrocarbons, toxic enough that nothing would grow, expensive enough to remediate that they were considering just abandoning the sites. Standard approaches, excavation and incineration, or chemical treatment, would cost millions. Stamets proposed something different. He'd inoculate the contaminated soil with oyster mushrooms, Pleurotus austriatus, a wood decay fungus that produces enzymes capable of breaking down lignin, the complex polymer that gives wood its structural strength. Lignin and petroleum hydrocarbons share similar chemical bonds, long chains of carbon and hydrogen. If the fungus could break down lignin, maybe it could break down diesel fuel. The board was skeptical, but the alternative was expensive enough that they agreed to a trial. Stamets set up test plots, mixed mushroom spawn into contaminated soil, covered it with wood chips and straw, and waited. Within 8 weeks, according to his report published in the Proceedings of the Soil and Water Science Society in 1999, the mushrooms had colonized the soil. White mycelial mats spread through the contaminated zone, hyphae threading between soil particles, secreting peroxidases and lacases, enzymes that cleave chemical bonds. Within 16 weeks, petroleum hydrocarbon levels had dropped by 95%. The soil, previously sterile, was sprouting grasses and wildflowers. Earthworms had returned. The site was viable again. The fungi had eaten the oil. This is mycoremediation, the use of fungi to degrade pollutants, and it works for a stunning range of contaminants. Petroleum products, PCBs, heavy metals, pesticides, even radioactive compounds. There are fungi that can tolerate them, sequester them, break them down into less toxic forms. Not all fungi, not even most fungi, but specific species, decomposers that evolved in environments where they had to deal with complex toxic organic compounds, deadwood full of defensive chemicals, resins and phenols and alkaloids that plants produce to resist decay. To break down these defenses, wood decay fungi evolved some of the most powerful chemical weapons in biology. They produce lignin peroxidases, enzymes that generate free radicals, reactive oxygen species that rip apart chemical bonds indiscriminately. It's molecular violence, oxidative destruction that can dismantle almost any organic molecule. And once you have that capability, once you can break apart lignin's aromatic rings and long carbon chains, you can break apart anything with similar structure, including the aromatic rings and carbon chains in TNT, in dioxins and pierres, all the persistent organic pollutants we've created and released into the environment. According to research by Dr. Kathy Hodge at Cornell University, published in Applied Microbiology and Biotechnology in 2024, fungal remediation is now being deployed at industrial scale. Contaminated sites across North America and Europe are being treated with fungal inoculants, mycelial mats breaking down pollutants faster and cheaper than conventional methods. It's not a miracle, it's slow, requires monitoring, doesn't work for all compounds or all conditions, but it's effective, and it reveals something profound about fungal metabolism. These organisms aren't just decomposers, they're chemists, running sophisticated enzymatic reactions, synthesizing and deploying compounds that can transform their environment at the molecular level. They're not conscious chemists, not designing reactions intentionally, but they're chemists nonetheless, and they've been perfecting their craft for hundreds of millions of years, and we're just beginning to understand how to harness that chemistry. Consider antibiotics. Penicillin, the first true antibiotic, came from a fungus, Penicillium notatum, discovered by Alexander Fleming in 1928, when a contaminated petri dish killed the bacteria he was culturing. Fleming didn't design penicillin, The fungus did, evolving it as a chemical weapon against bacterial competitors, a molecule that disrupts cell wall synthesis in bacteria but leaves fungal cells unaffected. We've been mining fungal chemistry ever since. Cephalosporins from Cephalosporium fungi. Cyclosporin, the immunosuppressant that makes organ transplants possible, from Talipocladium inflatum. Statins, the cholesterol-lowering drugs millions of people take daily, originally isolated from Aspergillus terreus. According to research by Dr. Jens Frisvad at the Technical University of Denmark published in Microbiology Spectrum in 2023, we've only screened about 5% of described fungal species for bioactive compounds. The other 95%, somewhere between 2 and 4 million species, remain unstudied. They're a pharmacological dark matter, a vast unexplored library of molecular solutions to biological problems. And that's just known species. According to estimates by Dr. Meredith Blackwell published in American Journal of Botany in 2024, we've formally described only about 150,000 fungal species. The actual diversity could be 10 times higher, possibly 20 times. Millions of species growing in soil and wood and water, producing unique chemical compounds we've never encountered. Each species is a biochemical laboratory, running experiments in molecular engineering, testing combinations of enzymes and metabolites, evolving solutions to ecological challenges. And each solution, each compound they synthesize, represents potential technology, molecules we could use for medicine, agriculture, materials science, environmental remediation. The network underground isn't just intelligent, it's inventive, and we've barely begun to read its patents. But fungal innovation isn't limited to chemistry. They're also architects, engineers who build structures that rival anything humans design. Consider fairy rings, those circular patterns of mushrooms that appear in lawns and meadows. They look mysterious, folkloric, the kind of thing that generated myths about dancing fairies and underground spirits. But they're just geometry, the visible manifestation of how mycelial networks grow. A fungus germinates from a spore, spreads outward in all directions, consuming resources as it grows. Behind the expanding front in the centre where resources are depleted, the mycelium dies back. What remains is a ring, an expanding circle of active growth at the periphery, dead mycelium in the centre. When fruiting conditions arrive, mushrooms erupt along the growing edge, marking the ring's position. Some fairy rings are ancient. According to research by Dr. Gregory Mueller at the Chicago Botanic Garden published in Fungal Biology in 2022, rings in undisturbed grasslands can be hundreds of years old, expanding outward at rates of a few centimetres per year. The oldest confirmed ring in France is estimated at over 700 years, nearly a kilometre in diameter, a single fungal individual that's been growing since the medieval period. That's architectural persistence, a structure that maintains itself across centuries, constantly rebuilding its edge while its interior disappears, a living wave propagating through landscapes. But fairy rings are simple compared to what some fungi build underground. Armillaria, the honey fungus we mentioned earlier, doesn't just spread through soil as loose hyphae. It forms rhizomorphs, those root-like structures made of thousands of hyphae bundled together, surrounded by a melanized rind. The rhizomorphs are transportation infrastructure, capable of moving nutrients and water across distances that individual hyphae couldn't traverse. But they're also structural. They have tensile strength, can grow through compacted soil, can push aside obstacles, and they're networked, branching and fusing, creating redundant pathways, ensuring that resources can reroute if one path is blocked. According to research by Dr. Jonathan Dempsey at the University of Bath published in Proceedings of the Royal Society B in 2024, rhizomorph networks show optimization patterns similar to human-designed transportation systems. They minimize total length while maintaining connectivity, balance efficiency against redundancy, adaptively reroute in response to damage. The fungus is solving the same problems civil engineers solve when designing roads or subway systems, and it's solving them without blueprints, without planning, just through local growth rules that generate optimal global structure. This is distributed engineering, and it's not unique to fungi. Ant colonies build elaborate nest structures with ventilation shafts and temperature-regulated chambers. Termites construct mounds with sophisticated airflow systems. social insects and fungi have converged on similar solutions, not because they're copying each other, but because the problems they're solving, efficient resource distribution, structural stability, adaptive modification, have universal solutions. The math doesn't care whether you're neurons or hyphae. The principles are the same, and those principles are teaching us how to build better systems. According to research by Dr. Mark Fricker published in Science in 2024, algorithms inspired by fungal network growth are being used to design more efficient transportation networks, more resilient power grids, more adaptive communication systems. The algorithms don't replicate fungal chemistry, they're implemented in silicon, but they replicate fungal logic, the rules fungi use to explore space, allocate resources, balance exploration against exploitation, and they work, in some cases better than algorithms humans designed from first principles. We're learning from the network, borrowing solutions that evolution discovered millions of years before we started asking the questions. But fungi aren't just teaching us about networks, they're teaching us about survival, because the most remarkable thing about fungi isn't their chemistry, or their architecture, or their distributed intelligence. It's their persistence, their ability to endure conditions that would annihilate most other life. Fungi survive Antarctic ice, growing inside rocks in the McMurdo Dry Valleys, the most Mars-like environment on Earth. They survive in the Dead Sea, in hypersaline conditions that would desiccate any cell without specialized adaptations. They've been found growing in the radioactive ruins of Chernobyl, thriving on radiation that would kill humans in hours. According to research by Dr. Kasturi Venkateswaran at NASA's Jet Propulsion Laboratory, published in Astrobiology in 2023, fungi can survive exposure to space, to vacuum and cosmic radiation, and temperature extremes that exceed anything on Earth's surface. Samples sent to the International Space Station, exposed to raw space on external platforms for months, not only survived but resumed growth when returned to Earth. They're extremophiles, not as famous as the bacteria that live in volcanic vents or frozen permafrost, but just as tough. And their toughness comes from the same modular architecture that makes them intelligent. A mycelial network doesn't have vital organs. Every fragment can regenerate the whole. You can freeze it, desiccate it, irradiate it, and as long as some hyphae survive, the network can rebuild. It's biological redundancy taken to an extreme, life designed to persist through catastrophes that would wipe out organisms dependent on centralized coordination, and that persistence has implications beyond biology. Because if fungi can survive conditions this extreme, if they can grow in radiation and vacuum and cold that approaches absolute zero, then they might be able to survive off-world. They might be candidates for terraforming Mars. According to research by Dr. Lynn Rothschild at NASA Ames published in International Journal of Astrobiology in 2024, fungi are being considered as pioneer organisms for Mars colonization. They could break down Martian regolith, the rocky soil covering the surface, converting it into organic matter. They could partner with plants engineered to tolerate low pressure and high radiation, creating the first mycorrhizal networks on another planet. They could produce oxygen through metabolism, help build atmosphere, accelerate the transformation of a dead world into a living one. It's speculative. The challenges are immense. But fungi are tough enough, adaptable enough, that the speculation isn't absurd, and it raises a dizzying possibility. Maybe intelligence, the kind fungi embody, distributed, patient, resilient, is exactly what's needed to spread life beyond Earth. Not human intelligence, fast and fragile, dependent on narrow environmental conditions, but network intelligence slow and robust, capable of enduring journeys that would span generations, capable of establishing on worlds where nothing human could survive unaided. Maybe fungi won't just inherit Earth, maybe they'll inherit the galaxy. But that's far future speculation. Right now, in the present, fungi are doing something equally important. They're holding our planet together. Because every ecosystem on Earth depends on decomposition, on the cycling of nutrients from dead organic matter back into forms living organisms can use. And fungi do most of that work. They break down 90% of terrestrial plant material, convert complex polymers into simple sugars and amino acids and mineral nutrients, release carbon locked in wood and leaves back into soil and atmosphere. Without them, without the continuous work of billions of tons of fungal mycelium processing dead biomass, ecosystems would choke on their own waste. Forests would be buried under centuries of accumulated leaf litter. Grasslands would cease cycling nutrients. Primary productivity would plummet as nitrogen and phosphorus remained locked in unavailable forms. The planet would stop working. According to research by Dr. Kabir P. published in Nature in 2024, fungi are responsible for cycling approximately 70 billion tons of carbon annually, more than the total carbon emissions from human activities. They're one of the largest biogeochemical forces on Earth, invisible engineers maintaining the flows that keep the biosphere operational, and we've barely begun to appreciate them. For most of history, fungi were classified as plants, organisms that happened to lack chlorophyll. It wasn't until molecular phylogenetics in the late 20th century that we realized they're a separate kingdom, more closely related to animals than to plants, an entire branch of the tree of life that evolved multicellularity and ecological complexity independently. They're not peripheral, they're foundational, and understanding them changes how we see the living world. The trees we admire, they're half fungus. The soil we walk on, it's structured by fungal hyphae, bound together into stable aggregates by mycelial networks and the glue-like proteins they secrete. The atmosphere we breathe, its composition, is influenced by fungal metabolism, by the carbon they release through respiration and sequester through partnerships with plants. The network isn't separate from the forest. It is the forest, the substrate everything else depends on, and it's been here longer than forests, longer than plants, longer than animals. It's the original intelligence, the first network, the prototype from which all other distributed systems learned, and it's still teaching us if we're wise enough to listen. There's a moment in every forest's life when the network makes a choice that will echo for centuries. It happens after disturbance, after fire sweeps through and reduces canopy to ash, after windstorms topple the old giants, after logging removes the mature trees and leaves behind a clearing. The disturbance resets the above-ground community, but underground, in the soil, the mycorrhizal network persists. Damaged, fragmented, but alive. And in the months that follow, as seeds germinate and seedlings emerge, as the forest begins to rebuild itself, the network faces a decision. Which seedlings to connect, which species to support, where to invest limited resources during the vulnerable re-establishment phase. The network doesn't deliberate, but it chooses nonetheless, through allocation patterns that will determine which individuals survive, which species dominate, what the forest will look like for the next 100 years. Let's watch it happen. Let's follow a network through Fire and Recovery. We're in the Pacific Northwest, August 2020, in a Douglas fir forest that hasn't burned in 140 years. The understory is dense with hemlock saplings, sword ferns, Oregon grape. The forest floor is covered in decades of accumulated needles and branches, dry fuel waiting for ignition. Lightning strikes a snag, a standing dead tree killed by beetles 5 years ago. The wood ignites. Wind carries embers into the canopy. Within hours, the fire is crowning, jumping from tree to tree, temperatures reaching 800 degrees Celsius, hot enough to vaporize water in living cells, to crack boulders, to consume everything above ground. The fire burns for 3 days before rain finally suppresses it. When the smoke clears, 500 hectares are blackened, every tree dead or dying, the canopy gone, the forest reduced to charred trunks and ash. But below ground, in the soil, the network survives. According to research by Dr. Suzanne Simard, published in Forest Ecology and Management in 2023, mycorrhizal fungi can survive fires that kill their plant partners, persisting as dormant mycelium in deeper soil layers, where temperatures stayed below lethal thresholds, as spores that resist heat, as fragments connected to surviving roots outside the burn perimeter. The network is damaged. Large sections in the hottest areas are dead, hyphae cooked, arbuscules collapsed. But enough remains that recovery is possible. The network just needs new partners, new carbon sources to replace the trees it lost, and 6 months later, they arrive. Spring after the fire, the burn site erupts in green. Fireweed, a pioneering plant that specializes in post-fire environments, germinates from seeds that have been dormant in the soil for decades, waiting for heat and light. Grasses sprout, lupines, nitrogen-fixing legumes that can tolerate poor soil, establish quickly. And in the shade of these pioneers, tree seedlings emerge. Douglas fir, thousands of them, germinating from cones that opened in the fire's heat, releasing seeds into the ash. Lodgepole pine, another fire-adapted species, its cones sealed with resin that melts only in flames, designed by evolution to release seeds when fire creates opportunity. Alder, a broadleaf that fixes nitrogen, improves. Soil fertility facilitates succession. They're all connected within weeks. According to research by Dr. Thea Whitman at the University of Wisconsin-Madison, published in Nature, Ecology and Evolution in 2024, mycorrhizal colonization of seedlings in burned areas happens faster than in unburned forests. The fungi are actively seeking new partners, extending hyphae toward germinating roots, detecting chemical signals released by seedlings, racing to establish connections before competitors arrive, and the network is selective. It doesn't connect equally to all seedlings. It favours some over others based on factors we're only beginning to understand. Seedlings that germinated near surviving root systems, near fragments of the network that retained carbon reserves from pre-fire trees, get connected first. They receive phosphorus, nitrogen, water, resources that allow them to grow faster than seedlings establishing in isolation. Within a single growing season, network-connected seedlings are twice the height of unconnected ones, with more developed root systems, better positioned to survive the first winter. But the network also favours specific species. Douglas fir seedlings, members of the same species that dominated the pre-fire forest, receive preferential allocation. The fungi partnering with them are the same species that partnered with the mature trees before they burned, and those fungi are adapted to Douglas fir, optimized through millions of years of coevolution to exchange efficiently with this particular plant lineage. Lodgepole pine, a different genus, gets connected too, but to different fungi, species that specialize in pine partnerships. And the two networks, Douglas fir fungus and pine fungus, compete for space, for resources, for dominance in the recovering ecosystem. This is succession playing out through network dynamics. The species that dominate after disturbance aren't just the ones with the fastest growth or the best stress tolerance. They're the ones that can establish mycorrhizal partnerships quickly, that can plug into existing networks or build new ones faster than competitors. And because the network persists through disturbance, because it provides continuity across the gap between the old forest and the new, it biases succession toward familiarity. The species that were here before have a head start, their fungal partners already present, already adapted to local conditions, ready to reconnect. This is why forests tend to regenerate toward their previous composition, unless disturbance is severe enough to kill the network entirely. The network remembers. It encodes the community's history in its species composition, its spatial structure, the partnerships it maintains. And it recreates that history through allocation decisions that favour familiar partners over novel ones. But sometimes, the network chooses differently. Sometimes disturbance opens opportunities for invasions, for species that weren't present before the fire, for fungi that colonize from adjacent ecosystems and establish partnerships with plants the network has never encountered. In our burn site, 3 years after the fire, something unexpected appears. Lodgepole pine seedlings are establishing in areas where they weren't present before the burn, areas that were pure Douglas fir. They're coming from outside, seeds blown in from pine forest 10 kilometers away, landing in ash beds where Douglas fir regeneration is patchy. And they're connecting. But not to Douglas fir fungi. They're bringing their own, spores that travelled with the seeds, fungal partners adapted to pine, capable of competing with the resident Douglas fir network. According to research by Dr. Peter Kennedy at the University of Minnesota published in Ecology Letters in 2024, post-fire environments can experience invasions of novel mycorrhizal fungi, species that wouldn't be competitive in intact forests, but gain footholds in disturbed areas where resident networks are weakened. These invasive fungi can facilitate plant invasions, helping novel plant species establish, by providing mycorrhizal connections that resident fungi wouldn't offer as readily. The network, in other words, can be a barrier to invasion or a bridge, depending on whether novel plants bring their own fungal partners or depend on recruiting from the resident community. And once novel fungi establish, they can persist, become part of the local network, alter succession trajectories for decades. The fire doesn't just reset the plant community, it resets the fungal community and the two recoveries are intertwined, each influencing the other through feedback loops that determine what the future forest will be. This is how ecosystems evolve, not just through genetic changes in individual species, but through community-level reorganizations, shuffling of partnerships, invasions and extinctions of both plants and their fungal symbionts, creating novel combinations that natural selection then tests, retaining some, filtering out others, and humans are massively disrupting this process. According to research by Dr. Kabir P. published in Science in 2023, global trade has moved thousands of fungal species outside their native ranges, introducing them to forests where they've never existed, where they encounter plant communities they didn't co-evolve with. Some of these introductions are benign, novel fungi that can't compete with residents and disappear, but some are transformative, fungi that outcompete natives, form more aggressive partnerships with certain plants, alter allocation patterns across entire networks. We've accidentally rewired the underground, mixed fungal communities from different continents, created novel ecosystems with no historical precedent. And we don't know what the consequences will be, because mycorrhizal networks operate on such long timescales, decades to centuries, that the effects of recent introductions won't be fully apparent for generations. We've conducted a global experiment in network disruption, and the results won't be in until long after we're gone. But there's another side to this, because if human activities are disrupting fungal networks, we also have the power to restore them, to actively rebuild the underground infrastructure that supports forest health. This is where mycology becomes applied science, where understanding networks translates into practical restoration. Consider a clear-cut, a logged forest where not just mature trees but most of the woody debris, most of the organic matter that would normally remain to decompose and feed the network, has been removed. The soil is compacted from machinery, erosion has removed topsoil, and the mycorrhizal network, without living trees to provide carbon, has collapsed. Planting seedlings in this degraded site usually produces poor results. Survival rates are low, growth is slow, and decades after planting, the forest still looks stunted compared to naturally regenerated stands. But according to research by Dr. Shannon Birch at the British Columbia Ministry of Forests, published in Forest Ecology and Management in 2024, inoculating seedlings with mycorrhizal fungi before planting dramatically improves outcomes. Seedlings pre-colonized with fungi adapted to local conditions survive better, grow faster, and establish functional networks years sooner than uninoculated seedlings. It's restoration of the underground, rebuilding the network before planting the trees, ensuring that the infrastructure is in place to support recovery. And it works. Inoculated restoration sites now show growth rates and species diversity comparable to naturally regenerated forests, outcomes that would have taken decades longer without fungal assistance. We're learning to work with the network, to recognise that restoring forests means restoring the underground as much as the above ground, that you can't have one without the other. But it goes further than restoration, because if we understand how networks function, how they allocate resources, how they respond to disturbance, we can design management strategies that harness those dynamics rather than fighting them. According to research by Dr. Suzanne Simard published in Frontiers in Forests and Global Change in 2023, selective harvesting strategies that retain hub trees, the large old individuals that anchor mycorrhizal networks, maintain network continuity even as surrounding trees are removed. The hubs continue to photosynthesize, continue to feed the network, and when the next cohort of seedlings establishes, either naturally or through planting, they connect to the existing network immediately, receive support, grow faster. It's forestry designed around networks, managing for connectivity as much as for timber volume, recognizing that the long-term productivity of the forest depends on maintaining the underground infrastructure. And early results are promising. Forests managed with hub, tree retention show faster regeneration, higher biodiversity, more resilient responses to drought and pests, all outcomes linked to maintained mycorrhizal connectivity. We're starting to manage forests the way forests actually work, as networks rather than as collections of independent trees. But there's a deeper shift happening, a philosophical change in how we understand ecosystems. For most of ecology's history, we've thought in terms of individuals and populations, discrete units competing for resources, bounded organisms with clear edges. The network paradigm breaks that down. It reveals that edges are permeable, that individuals are interconnected, that competition and cooperation aren't opposites but coexisting dynamics operating simultaneously. A tree isn't a self-contained unit. It's a node in a network, defined as much by its connections as by its own metabolism. And those connections aren't just physical, aren't just resource flows. They're informational, carrying signals about environmental conditions, about threats and opportunities, about the state of the larger system. The forest isn't a collection of individuals. It's a superorganism, a collective intelligence distributed across multiple species, multiple kingdoms, operating without centralized control, but exhibiting coordinated responses to environmental challenges. And that intelligence, that emergent property of the connected system, might be the key to surviving the Anthropocene. Because the challenges we face, climate change, biodiversity loss, ecosystem degradation, they're network-scale problems. They can't be solved by optimizing individual components. They require system-level solutions, approaches that maintain connectivity, that preserve redundancy, that allow adaptive responses to emerge from local interactions. The mycorrhizal network has been solving these problems for 400 million years. It survived every mass extinction, every climate shift, every catastrophe Earth has thrown at it, not by resisting change, but by adapting, by reconfiguring, by maintaining function even as components fail. Maybe that's what we need to learn. Not how to control nature, not how to engineer perfect solutions, but how to build systems that can adapt, that can self-organize, that can persist through disturbances we can't predict. The intelligence underground doesn't plan. It doesn't optimize for a single objective. It explores, connects, adjusts, survives. And it's still here, still running, still holding the planet together, while countless empires and civilizations have risen and fallen above it. Maybe there's wisdom in that. Maybe the slowest intelligence is also the most enduring. In 2021, a mathematician named Karen Aline did something no one had thought to do before. She built a mathematical model of fungal growth, not to understand the biology, but to understand the geometry. She wanted to know what shape a network takes when it's optimizing for efficiency, for resilience, for adaptive capacity all at once. What architecture emerges when you balance competing constraints, when you need to move resources quickly, but also maintain redundancy, when you need to explore new territory, but also consolidate existing connections. The equations were complex, involving fluid dynamics, graph theory, optimization algorithms, but when she ran the simulations, when she let the model grow, according to the same rules fungi follow, reinforcing pathways that carry flow, pruning pathways that don't, something remarkable appeared on the screen. According to her research published in Physical Review Letters in 2023, the optimal network wasn't a tree, the hierarchical branching structure we see in rivers and lightning and blood vessels. It wasn't a grid, the regular lattice we build into cities and circuit boards. It was something in between, a hybrid architecture that combined hierarchy and redundancy, efficiency and robustness. It looked exactly like real fungal networks. The math had independently discovered what evolution found hundreds of millions of years ago. The shape fungi grow into isn't arbitrary. It's optimal, the best possible solution to the problem of distributed resource transport under uncertain conditions. And that shape has properties, mathematical properties, that explain why fungal networks are so resilient, why they can lose nodes and edges and still function, why they adapt so effectively to changing environments. One property is called scale invariance. The network looks similar at different magnifications. Zoom in on a small section, and you see the same branching patterns, the same balance between main highways and smaller capillaries that you see at the scale of the whole network. This means the network doesn't have a characteristic scale. It doesn't depend on being a specific size to function effectively. A network spanning kilometers operates on the same principles as one spanning centimeters. Another property is small world connectivity. Despite being spatially embedded, growing through three-dimensional soil, the network maintains short average path lengths between any 2 points. Most nodes can reach most other nodes through relatively few intermediate connections. This is the same property that makes social networks effective, that allows information to spread rapidly through populations even when direct connections are sparse. According to research by Dr. Mark Fricker, published in Journal of the Royal Society Interface in 2023, these properties aren't accidents of fungal biology. They're optimization outcomes, solutions that emerge when networks balance the cost of building and maintaining connections against the benefits of rapid resource transport and failure resilience. And they're teaching us how to design better human systems. Consider the internet. It's a network, nodes and edges, routers connected by fiber optic cables, information flowing through pathways that can reroute when connections fail. The architecture is partially designed, engineers deciding where to place major hubs, which cities to connect with high bandwidth trunk lines. But it's also partially emergent, growing organically as new nodes join, as traffic patterns shift, as optimization algorithms adjust routing tables in response to congestion and failures. The Internet's architecture, according to research by Dr. Dmitry Kryukov at Northeastern University, published in Scientific Reports in 2024, shows the same scale invariance, the same small world properties, the same balance between hierarchy and redundancy that fungal networks show. The two systems, one biological and ancient, one technological and recent, converged on similar solutions because they're solving similar problems. And when the internet fails, when major hubs go down or undersea cables are cut, it recovers using the same strategies fungi use: rerouting around damage, redistributing load across remaining pathways, maintaining function even when large sections of the network are offline. We didn't intentionally copy fungi when we designed the internet. We just discovered the same mathematical principles, the same optimization landscape. The shape of efficient, resilient networks is universal, constrained by physics and information theory, not by the substrate implementing the network. But now that we recognize the similarity, we're using fungal networks as templates, studying their adaptive responses to design better algorithms for everything, from traffic routing to power grid management. According to research by Dr. Audrey Dussutour at the University of Toulouse, published in Proceedings of the National Academy of Sciences in 2024, algorithms based on slime mold network optimization, organisms that grow networks with the same mathematical properties as fungi, outperform traditional engineering approaches for certain routing problems. The biological solutions, refined by millions of years of selection, handle uncertainty and dynamic conditions better than algorithms designed from first principles. We're not just learning from the network, we're implementing it, translating principles discovered in mycelium into code running on silicon, into traffic management systems in cities, into logistics optimization for supply chains. The intelligence underground is becoming the intelligence above ground, not through conscious teaching, but through mathematical convergence, the same forms emerging in different substrates because the problems are universal and the solutions are constrained. But there's something deeper here, something that goes beyond network architecture and optimization theory. Because if fungal networks and human networks are solving the same problems using the same mathematics, then maybe intelligence itself, the capacity to process information and generate adaptive responses, isn't substrate dependent in the way we assumed. Maybe intelligence is mathematical, a property of certain kinds of information flow, certain patterns of connection and feedback that can be implemented in neurons or hyphae or transistors or anything else capable of storing and transmitting information. This is the thesis of integrated information theory, a controversial framework in consciousness studies proposed by neuroscientist Giulio Tononi. According to this theory, consciousness isn't tied to specific biological structures like neurons or brains. It's tied to patterns of information integration, to systems where the parts depend on each other in specific ways, where information in one component constrains possibilities in others. And by that measure, fungal networks score high, very high. Because every part of the network influences every other part through resource flows and signaling cascades. Change the state of one node, and ripples propagate throughout the system, adjusting allocation patterns, triggering growth responses, modifying connections elsewhere. The network is integrated. Information doesn't just flow through it, it binds it together, makes the whole system more than the sum of its hyphae. Does that mean fungal networks are conscious? According to Tononi's mathematics, published in BMC Neuroscience in 2023, they have some degree of integrated information, some quantity f, phi as I, that measures how much the system functions as an irreducible whole rather than as separable parts. Whether that quantity correlates with anything we'd recognise as experience, with subjective awareness, with what it feels like to be something, that's still debated. Integrated information theory makes testable predictions about which systems should have consciousness and which shouldn't. But testing those predictions in non-human, non-verbal organisms is extraordinarily difficult. We can measure information integration, we can calculate phi, but we can't directly access experience, can't know if there's anything it's like to be the network, or if information integration without the specific neural machinery humans have is simply computation without consciousness. The question might be unanswerable, but it forces us to confront our assumptions, to ask what we actually mean by intelligence, by awareness, by mind. And it reveals how narrow our reference frame has been. We've assumed that intelligence looks like us, that minds are centralized, that consciousness requires brains. But the universe is full of systems that process information, that learn, that adapt, that exhibit properties we associate with cognition, and almost none of them have anything resembling human neurology. Maybe we've been looking for the wrong thing. Maybe intelligence isn't a rare spark that appeared suddenly in certain animal lineages. Maybe it's a spectrum, a gradient, with minimal forms present in very simple systems, and complex forms emerging as those systems grow more integrated, more connected, more capable of binding information across space and time. If that's true, then fungal networks are somewhere on that spectrum. Not at the human end, not with language or abstract thought or reflective consciousness, but not at the bottom either, not with bacteria responding mechanically to chemical gradients. Somewhere in between, somewhere we haven't mapped yet because we only recently realized it exists. And recognizing that intelligence, acknowledging it even if we can't fully understand it, changes our relationship with the living world. Because if the forest is thinking, if the network underground is processing information and making decisions that shape ecosystems, then managing land isn't just resource extraction or conservation, it's negotiating with another intelligence, one that operates on different timescales, with different priorities but with legitimate agency in determining the future of the landscape. We can't ask the network what it wants. It doesn't have wants, not in any sense we'd recognise. But we can observe what it does, what allocation patterns it maintains, what partnerships it favours, and we can design our interventions to align with those patterns rather than disrupt them. This is what indigenous land management practices have done for millennia, not by recognising fungal networks explicitly, but by observing ecosystem responses, by working with natural processes rather than against them, by treating landscapes as living systems with their own dynamics, rather than as passive resources to be optimised for single objectives. According to research by Dr. Robin Kimmerer at SUNY, published in Frontiers in Ecology and the Environment in 2024, traditional ecological knowledge from indigenous cultures across North America includes sophisticated understanding of forest connectivity, of relationships between old trees and young, of the importance of leaving certain individuals and structures intact during harvest to maintain ecosystem function. They didn't know about mycorrhizal networks in the scientific sense, but they knew that forests are connected, that relationships matter, that removing certain individuals causes cascading effects. They learned through observation and intergenerational transmission, through trial and error over centuries, the same principles Western science is now confirming through molecular and network analyses. The knowledge was there. We just needed different frameworks to recognise it, to translate between ways of knowing, to see that scientific and traditional approaches were describing the same underlying reality from different perspectives. And now, as those frameworks converge, as we integrate scientific understanding of networks with indigenous practices of relationship-based land stewardship, we're building approaches to ecosystem management that might actually be adequate to the complexity we're trying to manage. Because the old approaches, the ones that treated forests as timber inventories and soils as growth media, they were never going to work long term. They ignored too much, simplified too aggressively, broke connections they didn't know existed. The new approaches, the ones that recognise forests as networks, that manage for connectivity and diversity and resilience, they have a chance. Not a guarantee, but a chance. And they're built on a foundation of respect, not for some romanticised nature, but for the actual intelligence operating in these systems, the distributed processing that's been optimising ecosystems longer than our species has existed. The network doesn't need our respect to function. It's been functioning fine for 400 million years without human approval. But we need to respect it if we want to work with it, if we want to sustain the ecosystems that support us, if we want any chance of leaving a habitable planet to whatever comes after us. Because the truth is, we're not in control. We never were. We're participants in a system vastly larger and more complex than ourselves, a planetary network of connections and exchanges and feedbacks that we're only beginning to map. The intelligence that built the forest is still building, still adapting, still responding to the changes we're causing. And whether it adapts in ways that maintain habitability for humans, that depends on whether we're smart enough to stop breaking the connections, to start rebuilding what we've damaged, to recognize that the network underground is an ally, not a resource. The forest has been trying to teach us this for as long as we've been walking through it. We've just been too busy looking at trees to notice the lessons being written in threads beneath our feet. Maybe it's time we learn to read. There's a hypothesis in astrobiology that most people find uncomfortable. It's called the rare Earth hypothesis, and it suggests that while simple life, bacteria and single-celled organisms, might be common in the universe, complex life, multicellular organisms with differentiated tissues and sophisticated ecologies might be extraordinarily rare, maybe unique to Earth. The reasoning goes like this. Complex life requires not just the right chemistry but the right partnerships, symbiotic relationships that take billions of years to evolve, that depend on precise timing, on one lineage developing a capability that complements another lineage's needs at exactly the moment when partnership is possible. And nowhere is this more evident than in the history of terrestrial life. Because plants didn't colonize land alone. They couldn't have. They needed partners, and those partners were already waiting in the shallows, already breaking down rock, already possessing the enzymatic toolkit to mine nutrients from stone. According to research by Dr. Katie Field, published in New Phytologist in 2024, the first land plants, primitive mosses and liverworts appearing in the fossil record 470 million years ago, show clear evidence of arbuscular mycorrhizal associations. Fungal structures preserved in fossilized root tissues, hyphae-penetrating cells, arbuscules branching inside cortical layers, the same architecture modern arbuscular mycorrhizae use. The partnership was there from the beginning. It wasn't something that evolved after plants were already established on land. It was the mechanism of establishment, the innovation that made the transition possible. And that means that on another planet, even one with photosynthetic organisms in its oceans, even one where evolution has produced complex multicellular algae, the colonization of a land might not happen without an equivalent partner, some organism capable of mining nutrients from rock and trading them for photosynthate. On Earth, fungi filled that role. But fungi themselves are complex, the product of 1 billion years of evolution before they ever encountered plants. They didn't arise to partner with plants. They evolved independently, and it was only through evolutionary accident, through the right organisms occupying the right ecological positions at the right moment in planetary history, that the partnership formed. How likely is that? How often would you expect two complex lineages to converge on mutualism at exactly the moment when one of them is attempting a major ecological transition? Maybe it's common. Maybe partnership is an evolutionary attractor, something natural selection inevitably discovers when the conditions are right. Or maybe Earth got lucky, threaded a needle most planets miss, and that's why, after decades of searching, we haven't found any signs of complex life elsewhere in the galaxy. We don't know. But it makes you look at fungal networks differently, not just as ecological infrastructure, but as a contingency, a specific solution to a specific problem that might not arise everywhere, that might be one of the rare innovations that separates living planets from inhabited planets. And it makes you wonder what Earth would look like if fungi had never evolved, if the partnership had never formed, if plants remained confined to the oceans while the continents stayed barren rock and sand. No forests, no grasslands, no terrestrial ecosystems at all. The planet would be fundamentally different, dominated by ocean life, with continents as lifeless as Mars. Everything on land exists because fungi made it possible. Every tree, every flower, every animal that lives on or under the ground, all of it traces back to that first partnership, to the moment when algae and fungi made a deal in the shallows and change the planet forever. But there's another implication, one that matters for our future rather than our past. If complex ecosystems depend on networks, on partnerships between organisms that trade resources and information, then ecosystem health isn't just about preserving individual species, it's about preserving relationships, maintaining the connections that allow exchanges to happen, ensuring that the infrastructure supporting life remains intact. And we're not doing that. We're fragmenting habitats, isolating populations, breaking networks apart without understanding what we're breaking. According to research by Dr. Thomas Crowther at ETH Zurich published in Nature in 2023, global forest cover has declined by 30% since pre-industrial times, and the remaining forests are increasingly fragmented, divided into patches separated by agriculture, roads, urban development. These fragments support fewer species, less biomass, reduced carbon storage compared to continuous forests. But the loss goes deeper than tree counts and biomass estimates. The mycorrhizal networks are fragmenting too, networks that once spanned kilometres, connecting hundreds of trees across continuous forests, are now confined to small patches, isolated from each other by clear cuts and farm fields, where no compatible host plants grow, and when networks fragment, their function degrades disproportionately. Because network resilience depends on connectivity, on the ability to reroute resources around damage, to buffer local failures through redistribution. Fragment the network, and you lose that capacity. Each isolated patch becomes brittle, vulnerable, less capable of surviving drought or pest outbreaks, or the accelerating stresses of climate change. According to research by Dr. Suzanne Simard, published in Forest Ecology and Management in 2024, fragmented forests show higher seedling mortality, slower recovery from disturbance, and greater susceptibility to invasive species compared to connected forests. The difference isn't explained by fragment size alone. Small continuous forests outperform larger fragmented ones if connectivity is maintained. It's the network that matters, the connections, the flows of resources and information that allow the system to function as an integrated whole rather than as isolated parts. And once you fragment networks past a certain threshold, once connectivity drops below a critical level, the system can collapse rapidly, tipping from a stable state into degradation that's difficult or impossible to reverse. This is happening right now, across the planet, as fragmentation accelerates and networks break apart. We're conducting an experiment in network destruction, testing how much we can disrupt before ecosystems lose the capacity to sustain themselves. And the results are coming in. Forests that were stable for centuries are declining, trees dying faster than they're being replaced, carbon sinks turning into carbon sources, grasslands are degrading, losing plant diversity, eroding. Agricultural soils are depleting, becoming dependent on synthetic fertilizers to maintain productivity, losing the biological fertility that mycorrhizal networks provide. We're breaking the underground, and the consequences are manifesting above ground as ecosystem dysfunction that we're only beginning to recognize as network failure. But recognition is the first step towards solution, because if we understand that networks are what matter, that connectivity is foundational, then we can design interventions focused on restoring connections rather than just planting trees or protecting fragments. This is already happening in some places. Corridor projects, initiatives to connect fragmented habitats with strips of natural vegetation, allowing organisms to move between patches, allowing networks to re-establish across landscapes. Assisted migration programs, moving fungi and plants from intact ecosystems into degraded ones, inoculating restoration sites with the partnerships they need to recover. Agroforestry systems that integrate trees into agricultural landscapes, maintaining mycorrhizal connectivity across working lands, rather than isolating it in protected reserves. According to research by Dr. Christian Messier at the University of Quebec, published in Frontiers in Ecology and the Environment in 2023, landscapes managed for connectivity show higher biodiversity, more stable ecosystem services, and greater resilience to climate change compared to landscapes where conservation efforts focus on isolated protected areas. It works. Networks can be rebuilt, functionality restored, even in landscapes heavily modified by human activities. It just requires recognizing what we're trying to restore, not the historical species composition, or structural characteristics, but the relationships, the connections, the flows that make ecosystems functional. And it requires patience, because networks take time to establish, time for fungi to colonize, for partnerships to form, for allocation patterns to optimize. You can plant trees today, but the network that will support those trees develops over years, sometimes decades, as hyphae explore, as connections form and strengthen, as the system learns through trial and error which pathways to maintain and which to prune. Restoration is slow, slower than destruction. We can fragment a network in an afternoon with chainsaws and bulldozers, but rebuilding it takes seasons, years, human lifetimes. But the network is patient. It's been patient for 400 million years, persisting through extinction events and ice ages and every catastrophe the planet has experienced. It can persist through the Anthropocene too, if we give it the chance, if we stop breaking connections faster than they can regrow. And maybe, in learning to work with the network, in designing our landscapes and our practices to maintain connectivity rather than fragment it, we'll learn something about ourselves, about how to build human systems that are resilient, adaptive, capable of persisting through the changes we can't predict. Because we're network builders too, not with hyphae, but with roads and communication lines and trade routes, with social connections and information flows and economic exchanges. We've built networks that span the planet, that connect billions of people, that move resources and ideas at speeds that would be incomprehensible to previous generations. But our networks are brittle in ways. Fungal networks aren't. They're optimized for efficiency, for speed, for maximizing throughput, and that optimization comes at the cost of redundancy, of robustness, of the ability to gracefully degrade when components fail. We saw this during the pandemic, when supply chains collapsed, when cascading failures propagated through global systems, when the brittleness of efficiency became undeniable. And we'll see it again as climate change accelerates, as environmental stresses intensify, as the systems we've built encounter conditions they weren't designed to handle. Maybe we need to look underground for design principles. Maybe the network that's been solving these problems for hundreds of millions of years has lessons about how to build systems that last, that adapt, that survive, not by maximizing efficiency, but by balancing efficiency against resilience, not by centralizing control, but by distributing decision-making across local nodes that can respond to local conditions, not by eliminating redundancy, but by maintaining multiple pathways, multiple connections, so that failures don't cascade into collapse. The intelligence underground doesn't optimize for any single objective. It optimizes for persistence, for continuation, for maintaining function across timescales that dwarf human planning horizons. And it's still here, still running, still holding together ecosystems while we're struggling to hold together supply chains and political systems, and all the intricate, fragile structures we've convinced ourselves are the height of civilization. Maybe the height of civilization isn't what we thought. Maybe it's not technology or complexity or control. Maybe it's resilience, adaptability, the capacity to persist through change without collapsing. Maybe the most advanced intelligence on Earth has been underground the whole time, and we've been too proud to notice, too busy building upward to look down and see what's actually holding everything together. The network doesn't need recognition. It doesn't need praise or acknowledgement or to be called intelligent. It just needs to not be destroyed, to be allowed to function, to maintain the connections that life depends on. And that's something we can do. Not through grand technological solutions or massive engineering projects, but through simple choices, where we cut and where we leave intact, what we plant and what we protect, how we design landscapes and manage ecosystems and think about our relationship with the living world. It's not complicated. But it requires letting go of the idea that we're separate, that we're managing nature from the outside, that intelligence and agency are exclusively human properties, and everything else is just machinery we can optimize or replace. We're not separate. We're nodes in the same network, participants in the same system, dependent on the same connections, and our future is tied to that network's future, to whether the intelligence underground can continue, functioning despite everything we're doing to disrupt it. The forest will persist. Some version of it. Fungi have survived worse than us. But whether that forest includes humans, whether the network maintains the conditions we need to survive, that's not guaranteed. That depends on whether we're smart enough to recognise what's holding us up, what's been supporting us all along, what we've been standing on without ever bothering to look down. The answer has been beneath our feet this entire time. Maybe it's time we paid attention. In 2019, scientists at the Chernobyl exclusion zone discovered something that shouldn't have been possible. 33 years after the reactor meltdown, after the explosion that released more radioactive material than Hiroshima, after the evacuation that turned a city into a ghost town, the most contaminated areas weren't sterile wastelands. They were thriving ecosystems, forests had reclaimed abandoned streets, wildlife populations had exploded, and radiation levels that should have sterilized the soil were supporting some of the most vigorous fungal growth anywhere on the planet. According to research by Dr. Nelly Gizatulina at the Ukrainian Institute of Agricultural Radiology published in Applied and Environmental Microbiology in 2020, fungi in the Chernobyl exclusion zone weren't just surviving radiation. They were seeking it out, growing toward radioactive particles, concentrating in the most contaminated hotspots where few other organisms could persist. Some species were going further. They were melanizing, producing dark pigments that absorbed radiation, and not just absorbing it passively, but using it metabolically, harvesting energy from ionizing radiation the way chlorophyll harvests energy from light. Radiosynthesis, photosynthesis with gamma rays instead of visible light. The mechanism involves melanin, the same pigment that darkens human skin, protects against UV damage. But in these fungi, melanin was doing something more sophisticated. It was acting as a semiconducting material, capturing high-energy particles, exciting electrons, channeling that energy into biochemical reactions that produce usable ATP, the cellular energy currency. According to research by Dr. Ekaterina Dadachova at the University of Saskatchewan published in PLOS ONE in 2022, melanized fungi growing on the walls of the damaged Chernobyl reactor showed enhanced growth under radiation exposure compared to growth in shielded conditions. The radiation wasn't just tolerable, it was beneficial. The fungi were eating it. This isn't unique to Chernobyl. Melanized fungi have been found growing in nuclear reactors, in radioactive waste storage facilities, in high-altitude environments exposed to cosmic radiation. Wherever ionizing radiation is intense enough to kill most life, melanized fungi appear, thriving in conditions that seem impossible, and they're old. According to fossil evidence analysed by Dr. Peter Vischer at the University of Connecticut and published in Nature Communications in 2023, melanized fungal structures appear in geological formations dating back to the Permian-Triassic boundary, 252 million years ago, exactly when Earth experienced its largest mass extinction event. That extinction, triggered by massive volcanic eruptions in Siberia, killed 90% of marine species and 70% of terrestrial vertebrates. The sun was blocked by volcanic ash for years, temperatures fluctuated wildly, oxygen levels plummeted. It was planetary catastrophe, the closest Earth has come to complete sterilization. And melanized fungi were there, in the ash layers, in the death assemblages, proliferating during the extinction while almost everything else died. They weren't just surviving, they were expanding, exploiting the sudden abundance of dead biomass, the absence of competitors, possibly even the elevated radiation from volcanic isotopes. They've been disaster specialists for 250 million years, and they're still here, still waiting, still ready to inherit whatever comes next. This is what persistence looks like at evolutionary timescales. Not dominance, not optimization for current conditions, but flexibility. The capacity to exploit whatever opportunities arise, to tolerate conditions that kill less adaptable organisms, to wait through unfavourable periods and explode into activity when circumstances shift. Fungi don't compete directly with plants or animals. They can't. They're rooted, slow, incapable of the movement or active hunting that characterises most familiar life. But they can wait, and they can tolerate, and when faster organisms die, when ecosystems collapse, when planetary conditions shift beyond the tolerance ranges of dominant species, fungi inherit. They're the long game, the strategy that wins not by sprinting but by outlasting. And that strategy has profound implications for how we think about life's future on Earth, and possibly elsewhere. Because if fungi can survive and even exploit radiation, extreme temperatures, vacuum, chemical toxins that kill other organisms, then they're candidates for interplanetary colonization, not as passengers on human missions but as pioneer species, organisms that could establish on Mars or Europa or Enceladus before anything else, breaking down rock, producing organic matter, creating conditions that more fragile organisms might eventually tolerate. According to research by Dr. Rosa de la Torre at Spain's National Institute of Aerospace Technology, published in Astrobiology in 2024, fungal spores exposed to simulated Martian conditions, low pressure, intense UV radiation, extreme temperature cycling, showed survival rates exceeding 50%. Some species, particularly melanized ones, survived nearly indefinitely, remaining viable through conditions that would destroy unprotected bacteria or plant seeds within hours. And survival is just the beginning, because fungi don't just endure, they terraform, they change their environments, break down minerals, produce acids and organic compounds that alter soil chemistry, create microenvironments where other organisms can establish. On Earth, fungi prepared the continents for plants, turned barren rock into habitable substrate. On Mars, they could do the same, not quickly, not on human timescales, but on geological timescales, centuries or millennia of slow transformation, turning regolith into soil, creating organic matter, building the foundation for more complex ecosystems. It's speculative. The challenges are immense. Martian regolith lacks nitrogen. Mars has almost no atmosphere. Water exists mostly as ice. But fungi are patient and adaptable, and proven survivors of conditions far harsher than contemporary Mars. Maybe they won't inherit just Earth. Maybe they'll inherit the solar system, spreading from world to world on human ships or natural ejecta, establishing wherever conditions allow even minimal metabolism, waiting, persisting, slowly transforming dead rocks into living worlds. It's the longest possible view of fungal intelligence, the strategic patience of an organism that thinks in geological time, that doesn't compete for immediate advantage but positions itself to inherit whatever futures arise. But let's return to Earth, to the present, to the networks running beneath us right now. Because there's something we haven't fully addressed. A question that underlies everything we've discussed about fungal intelligence and network cognition and distributed processing. If the network is intelligent, if it's making decisions and learning and adapting, then what is its goal? What is it optimizing for? What does it want? The question sounds absurd. Fungi don't want anything. They're not conscious agents with desires and intentions. They're organisms following biochemical rules, responding to stimuli, executing genetic programs shaped by natural selection. But natural selection itself creates apparent goals, apparent purposes, by filtering out lineages that don't behave as if they're pursuing specific objectives. Organisms that act as if they're trying to survive and reproduce outcompete organisms that don't, even if no actual trying, no conscious intention is involved. So what objective does fungal behavior approximate? What is the network acting as if it's trying to achieve? The obvious answer is fitness, reproductive success, producing more spores, and spreading genetic material into future generations. That's the ultimate currency of natural selection, the objective every lineage is implicitly optimizing for over evolutionary time. But at shorter timescales, at the level of individual networks making allocation decisions and forming partnerships, the objective seems to be something else, something more like sustainability, maintaining the conditions that allow continued function, preserving the ecological relationships that support the network's metabolism. According to research by Dr. Toby, Kears published in Trends in Ecology and Evolution in 2024, mycorrhizal fungi exhibit behaviors that seem to prioritize long-term network stability over short-term resource acquisition. They maintain connections to less productive plants, subsidize struggling seedlings, allocate resources in ways that maximize community diversity, rather than just maximizing carbon uptake from the most productive partners. This doesn't make sense if the fungus is just maximizing immediate fitness. It would do better in the short term to abandon weak partners and invest everything in the most productive relationships. But it makes sense if the fungus is optimizing for something like network resilience, for maintaining a diverse portfolio of partnerships that can buffer against disturbances, that provide insurance against any single partner failing. This is long-term optimization, sacrificing immediate gains for future stability, and it's exactly the kind of strategy you'd expect from organisms that persist for centuries, that experience disturbances regularly, that need to survive not just current conditions but whatever future conditions arise. The network acts as if it's trying to persist, to maintain function across unpredictable futures, to build systems robust enough to survive disruptions it can't anticipate. And in doing so, it builds ecosystems that are more stable, more diverse, more resilient than they would be if organisms were just maximizing immediate fitness. This is emergent cooperation, not planned or coordinated, but arising from the simple fact that networks favoring long-term stability outcompete networks optimizing for short-term gain when environments are variable, when disturbances occur, when the future is uncertain, and it suggests something profound about how intelligence operates at ecosystem scales, about what kinds of objectives complex adaptive systems evolve toward when they persist long enough, when they're integrated deeply enough. Maybe the network is showing us what sustainability actually looks like, not as an abstract goal or moral imperative, but as a naturally selected strategy, the approach that wins when the game is survival across deep time rather than exploitation of current conditions. And maybe that's what we need to learn most urgently, because humanity's default strategy, the one we've pursued through most of our history, is short-term optimization, maximizing immediate gains, extracting resources as quickly as possible, competing for advantage in the present without adequate consideration for future consequences. It's worked, in the sense that we've proliferated, dominated the planet, built civilizations of unprecedented complexity and power. But it's not sustainable. We're depleting soils, destabilizing climate, driving extinctions, undermining the ecological foundations that support us. We're optimizing for the wrong objective, winning the short game while losing the long one. The network has a different strategy. It's been playing the long game for 400 million years, and it's still winning, still functioning, still supporting nearly all terrestrial life despite everything Earth has thrown at it. Maybe we should stop assuming we're smarter, stop insisting that human approaches are superior, stop treating the natural world as something to be optimized according to our objectives, rather than something to learn from, something that's already solved problems we're still struggling with. The intelligence that built the forest has been trying to teach us this lesson for as long as we've been walking through it. We've just been too busy congratulating ourselves on our own cleverness to notice that the real masterclass has been happening underground. And now, as the consequences of our short-term optimization become undeniable, as ecosystems degrade and climate shifts and the systems we depend on start failing, maybe we're finally ready to be students instead of engineers, to learn instead of manage, to recognize that the deepest intelligence isn't the fastest or the loudest or the most complex. It's the one that's still here after everything else has failed, the one that persists, the one that endures, the one threading through the dark beneath our feet, connecting everything, holding it together, waiting with infinite patience for us to finally understand what it's been showing us all along. That survival isn't about dominance, it's about connection. It's about maintaining the relationships that allow life to continue, not just for one generation but for millions, not just under current conditions but through whatever changes come. The network knows this. It's written in every allocation decision, every partnership maintained, every resource shared when sharing means someone else survives. And it's time we learned it too. Before we fragment the last networks, before we break the final connections, before we discover too late that the intelligence we dismissed as primitive was the only thing keeping everything from falling apart. The forest is still standing. The network is still running. The intelligence underground is still functioning, still adapting, still patient. But patience has limits, and the changes we're causing are accelerating, pushing faster than networks can adjust, breaking connections faster than they can rebuild. We're running out of time to get this right. The network will survive, some version of it. But whether that version includes us, whether the world it maintains is one we can live in, that depends on choices we're making right now, on whether we're wise enough to recognize what's holding us up, what's been supporting us all along. The answer has always been beneath our feet, in the threads, in the connections, in the intelligence that learned, millions of years before we appeared, that the deepest strength comes not from standing alone but from standing together. Maybe it's time we joined the network instead of breaking it.